Hello, I'm chatting in the lapel mic. Hello? Testing, testing, testing. Stream test. Stream test.
They're doing my presentations in there for something else right now.
Oh, really?
Okay. Yeah, I don't know if that's part of their. You want to grab the clicker really quick? Just whenever they do like the virtual thing, I push the button. I'll be back in a minute. Yeah, we're streaming now. We just haven't pulled it up there. But we can get in there whenever. I don't know if they're still practicing.
We both said it was very nice.
I think we should have used it, guy.
It came out very nice.
Hey, Smiley and Savannah, can you guys send me your presentation? Where did you already?
I think we accidentally sent it as a PDF the first time, but we're gonna resend it right now as a PowerPoint file.
Oh, excellent. That
Yeah.
would be wonderful.
Yeah.
I think I have all of everybody's except for yours.
Oh.
Not to call you out.
Just your email?
My email would be wonderful. Thank
Gotcha.
you.
Oh yes,
Special.
it's right. Yeah, so we can see like the stream and stuff. Like, we don't really see. So you're telling me. And then he's gonna email me some presentations with the logo on it. More about PowerPoint.
Actually, I'm also missing Evelyn Gutierrez, her poster.
Okay.
Could you have her send it as well? Do you have contact with her?
Do you ever know?
I'll need hers.
And then what about you?
You said her poster?
Her oral presentation.
She's actually really fun though. That's pretty sure. I think that's done. Yeah. I think all of our stuff. Tell me. Just be on it stream. They will always be around. True.
Small.
You want some check out?
You say I'm gonna laugh at this.
It's not out of the start. Yeah.
It's gonna put me out of the job. Why? No, I'm just kidding.
just everyone's just joking on random. Because Chipotle was the first one. That's so awesome, dude. I've never been more excited to do something in my life. We should do. I might do it for the joke. Honestly, it's so fun. I might have to start doing it to like people for some stuff, some media. Benji, she wants that first graphic up as people are walking in.
this is another building. So we're near the end.
Yeah, great, fantastic, I love that. So let me see
No.
what is going on with this.
Okay, you want to
So I will be here, yeah?
I think so. I'm trying to see which computer is.
No, he will, I was told that the person will come, everything is in, I send in to Ali my stuff. I even have for emergency a stick, but
Yeah, that is, I'm trying to see which, there's two computers, I'm trying to see which one is running it.
yeah. But we will see, I mean, we still have lots of time.
A little bit of time.
Where do you have the restroom?
Yes, I will show
Because
you
we
that
will-
because I need to talk to these people in the back
Hi.
anyway.
I'm gonna go check on Jack. Sorry, check the last tables, but when if I get that email, I'll send it over. Got it.
Take notes.
Yeah. I already took statistics.
So you're professional.
Did Dr. Fuller get connected with Dr. Zimmer? Okay.
I retained that much information.
I just know it didn't happen sometime. That's so I got some random motion.
Oh, so that's another?
Or is it like one Jack and Howard? Yeah. I don't forget. How you doing?
Hello.
How are you doing?
What are you guys doing in here?
Bored.
Hanging out.
Bored?
Nothing.
Check out any posters?
Yeah, we
Okay.
did our rounds.
Did your rounds?
Yeah.
Alright.
So do you know where these slides are, like which computer is running it?
I have no idea.
Okay, that's what we're trying to figure out.
I'm gonna go meet Betty. She's upstairs.
Cause
Why are you doing that?
why am I doing it? Cause I was the first one to greet him and nobody else was in the room.
Ryan's his,
Ryan's
liaison.
his liaison?
Yep.
Okay.
I just,
Well, I
Anything
was
that's-
just here.
anything that's not my job, I
This
just back
is
off.
my thing.
Are you doing something here? Are you?
Well, I did posters at 8:30, I greeted him. 8:45, I gotta be back in Staples.
Oh,
9
that's your
o'clock, I'll
own,
be here.
that's your own, internal agenda.
This is my,
It's a lie.
It's a lie. That's not your alley generated agenda. I have my alley generated
no,
agenda.
this is
that I don't have my own agenda.
my jumping between,
Yes.
Park and James agenda.
Where is our tonight?
Staples and James Complex.
Staples. Okay.
So I've gotta hop back and forth. So, in fact, if everything is, you think everything is set here, I will hop back over there and make myself known and
You go make yourself done.
Okay, I just wanna, they have the 8:45 off original. What do you get? And this is Kendra. Yeah, perfect. Just wanna make sure. Allie, I don't wanna grab a program now, but can you save me one? Yes. Later. Any calls? Anytime.
This is our team who help you with your slides. You have Dr. Fly's slides, correct? Yes. Do you like? They're up right now. And we advanced the triangle slides. This is yes. This is wonderful. And then if we do the clicker for him. I think it might be coming from.
Just set up here.
Yeah. You introduce me.
I'll be introducing.
Clicker's up there, but let me hear you. Yeah, just tell me how to use it. Yes, let's go look for it.
Those two fellows sure have parts of
Oh, no, no. I haven't done much. It's all Ali and Laura and the team.
So here's your mic.
Yeah.
So
Do we need that? Yeah.
would you rather use a handheld? You can use either
No,
one.
it's fine.
Okay, so
We have people in another room. We have overflow to another room. We have 400 people who have signed up to come today, so this room holds 300, so we're gonna,
Wow.
We have 100 more.
So
in
just
there.
like right
Up to
here
100 more.
on your tie, or on the other one, other side here. Probably like right there. Yeah, and then you can just like put this in your jacket, inside maybe, and then this one. Sorry, I didn't grab it from you.
Mhm.
This
And
is
this one is,
forward and this is back. This doesn't do anything.
and the
forward.
pointer,
Mhm.
pointer is where?
It's broken.
Here, I can get you a pointer.
We
I
don't
have
have
a
a
laser I can-
working clicker that has a laser on it.
do you have one?
Yes, I'll get one really fast.
So it's forward like that, yeah?
Yeah.
And the pointer will be a separate one.
Yeah.
Now we are already on the second slide here.
Good morning.
Hi, Tom
Good.
Franco.
Yeah.
I'm Margaret
Want to test your mic?
Nice to meet you.
Turn it on real quick.
Yeah.
If I
Thank you.
I have this, and it's forward and
A
advance.
group of us, I'm associate
You have
dean
a-
in our natural
This
mathematical
has a
sciences. I
range
think
of
you were here
75
whenever
feet, so
we had a lunch
A lot of
couple
range?
years
yeah.
ago.
Can we
Yes,
try
yes.
it?
First set up.
We can try
Yes,
it.
yes,
And then
yes.
at least you could have the pointer.
but,
Yes,
and
the
forward.
pointer is
As we
important.
drove
Yes.
in
Forward,
today,
backward, that.
further
Here,
in.
why don't, well, here, let's plug it in and
we
let's
realized
try
that
it.
there's
Yeah,
some
Yeah, you
variation
can
of
turn it
this
on real
one.
quick.
Okay, yes,
In there.
And I just
A
talked
little
to
switch on top.
Rebecca,
yes.
and she told me about, there is a
This
place where we have old citrus
Excellent.
Hello?
Oh, yes. Oh, yes.
Hello?
and the program,
Yes.
the origin of
Try again.
citrus.
Hello?
Well, there's
It's good?
citrus
All right,
trees
so
in
keep it on.
the very
Alright, I'll
first
mute it
one
now. You
tables.
can just leave it on.
And then we have a grove here on campus.
Yes, and that's,
Thank you very much.
And then
this
this one is
closer
fine.
than
Yeah.
Well, here, let's try
Yeah,
this one out so
yeah.
you can get the pointer.
Sure.
Can we plug it in over here really quick and try it?
what's going on
Okay,
there.
excellent.
He's very interested because we have a lot of citrus
Leave
trees
it.
in our
Here,
backyard.
why don't I trade you?
I learned this
You
one.
take that and we'll.
I learned this
We started a
one.
course
Yeah.
in general education.
This one here
That's,
would be
yeah,
forward,
Looking at like
Yes.
history
or
of citrus
forward,
and
yeah,
citrus
that's what
residue.
Yes,
Rebecca
and then this
Go to
mentioned.
the
is your
citrus,
pointer.
Yeah.
It has an on-off switch. Right
students
here.
learn about
just-
propagating
just-
citrus
well,
and
Just
all
keep
that
it in mind.
He's so
stuff.
interested
So
in
you can
that
see.
because he grew
Yeah.
up
Hopefully
in
that's
a
bright
farm,
enough
not
for
with citrus,
the large
but,
room.
in Europe,
If not, I think we can deal with this.
Yes.
Yeah, and I can stay here anywhere.
Nowhere near what UCR's
Yes.
So
citrus
it looks
is,
like
but
that's,
ours is like
yes,
probably
that
student,
is
like
working
the
here
students
too,
get to
if
do
you wanna
all.
try it.
The only time
perfect?
we ever had something.
Yes,
like
I think
this
that's
were,
working just fine.
all the variations
The
and
Logitech?
the hybrids of the
You know what?
citrus.
Is it big enough
That's
from the
not
back? Can you
range.
see it?
Okay.
They do
Sorry?
that too,
Yes.
so they
Yeah.
experiment a lot.
Can you
Yes.
see it?
The
Let me,
pointer?
yeah, I can go to the, here, let me see if I can see that over here.
Yeah, I think you can see it.
Yes,
It's too many.
I think it's very
I get
visible.
so
Over there
frustrated when we just
No,
go
not over there,
sit
but
there and think
I
about,
I think even from the other side of the room, you
Good,
can-
it's fine.
it's very bright actually.
talks to his students in
Thanks.
Würzburg.
Yeah, it has a longer range, or?
It's a very,
Well, this is like 300, I
it's
think,
a restless life.
but
Yes.
yeah, that was probably like
Your mind
15
is
bucks.
constantly
This is like
turning,
300.
right? So.
Oh, wow.
I don't see the
All
worth
the
in
time.
it.
I'm
Because it doesn't
totally
have the
lost
laser.
because
When I'm in
that
my,
is
my
has
larger
his
classes
iPad,
in
but he
here, I'll
doesn't
usually
really
have it
want
plugged in the
to
front,
use
and I can walk all the way outside,
computers.
and I can still click. So
Yeah.
that's good enough for me, you know, maybe not
Oh,
for a
yeah.
larger
Well,
auditorium.
yeah, you need this for like graduation.
Yeah.
Then I have
Yes,
to
You don't
absolutely.
need this one anymore. It
scan
doesn't work
something.
Oh,
anymore.
you're his
But
assistant.
that one will
I'm
be,
his assistant.
will be
Yes.
more.
Okay.
Yes.
Like it has a laser on it.
See, I'm really.
It just
just
doesn't
doesn't
work.
work, yeah.
My own thing.
Yeah.
So, and I am fine with this.
Yes, I think
Well,
so. Well,
yeah.
this one is muted now, but we'll unmute it. When
Yeah,
we-
good.
But they'll take care of that for you, right? You can take care of it from the back? Excellent.
Reason
Hey,
that
thank you very
in
much.
the United States now for 50
Great. Is there anything we can do? Can I get you some water maybe? or-
Yes, please, a glass of water, no ice. Yeah.
Yes, absolutely.
thank you.
Could I get you a bottle? Would that be fine?
Yeah.
Be right back.
Projection of a lecture
Mm,
that a Nobel Prize laureate from California gave in Germany.
okay.
And yeah, you gave you set up the projection for Dr. Dalberg in Germany, and then he carried a suitcase. And my husband asked him, no, I'm Hungarian, and Hungary is my little, and they can
There's plenty.
let me get
As many as
outside
you
of
need.
the country.
Yeah, yeah.
And let's see how much time we have.
Scholarship.
Ah.
How is it? Can I get a
About 20
scholarship?
minutes.
He would say,
We could, I could let you see some of the posters, or if you'd like to just.
Yeah, I will. I
Rest
just
after.
talk to
just send me your thesis was left. conversation till our arrival in LA. 4 years went by. But when we came here and had a wonderful
Yeah, we were
4 years
very,
and
very
got,
lucky.
yeah,
50 years ago we started our
Hi.
house.
Yeah.
Ryan.
yeah.
It's amazing,
Yes.
huh?
Yes.
It's amazing how things happen.
I think we met
Yeah.
at
Is-
Loma Linda
is
with
your
Dr.
origin
Langridge
also from
when
our
you
corner?
visited with
You
your
wanna
grandson.
Slovakia.
Maybe like a year
Yeah,
or two
just
ago.
just north of your corner.
Yes.
Yeah.
What's your connection with language?
I actually worked in, I collaborate with his lab.
Yeah, all one
Still
country.
here at CU, but when I was a
Yeah.
graduate student there
Yes.
a few
Are
years ago. Yeah,
You were graduated with Bill?
not with him, but in the lab right next to him. And so I would spend a lot of time. He was on my committee, and, a dear friend of mine.
Bill was with us at Ithaca, New York.
Uh-huh.
Then with his family, my daughter was babysitting his young
Oh, really?
sons way back in Ithaca. She was a teenager.
Oh, wonderful.
And then we all moved to Edmond, Alberta together.
Yes,
And
yeah.
from there to Loma Linda.
Oh, you guys moved to Loma Linda with him then?
He moved with him.
Oh, yeah. Amazing, amazing.
Yeah,
So
from
he started.
Ithaca,
Is he from
Edmonton,
Slovakia?
Loma Linda. Always, he always followed
Yeah.
my husband.
And what was after Loma Linda? Did you stay in
Margo.
California?
Margo.
Did you move
We
on?
live here,
Okay.
Yes.
I'm gonna when I introduce.
but
you call
also,
that
yeah,
stuff.
he's also involved in those war.
Yes, yeah.
But I am very much encouraging him
Yeah.
to stop it and have some time, some more free time.
Yes,
Well, we're thankful that you're here and
We like to travel
your donation of the balance as well. We have
Whenever we
that. I
did
don't know if you've seen where that
traveling,
is
it's
outside of our chemistry area, and then
We give
just
lectures
being a
and
friend of
I,
ours and being open to
enjoy the
sharing with our
scenery
students
and
what
Yeah.
you're
we
what you're
were just
working
in-
on. So it's
in
a blessing to
Dubai
have you
and
here.
I took the tours and all this and he had that only a few hours and get the highlights and
That's fun.
yeah,
I look forward to doing that with my wife too sometime.
traveling is so
Yeah.
important. It opens up so many divisions of the world and understanding for people. When we were from Dubai, he has in
Yeah.
Würzburg, he has an associate from a lady from Egypt,
What you've been able to do.
Cairo.
Yeah.
And she said, once you're in our vicinity, why don't you then pop over from Dubai to Cairo? So we did and saw the pyramids and the museums and all this was wonderful. But always be together with local people. And I asked a lady who also associate somebody with somebody with the same who has the same interest he has in science. I asked her, where do local people eat? Where do they eat? What? And then she took us to an Arabic restaurant. There wasn't even English, anything
Yes. Well, our whole focus in
readable
this
to
event
us on the menu.
is give our students, our
Totally
undergrads, an opportunity
in her
What do we do?
hands.
to get involved in research.
And those experiences are really great. You should just nurture your scientific connections to people.
Because a lot of our students have no idea. They don't.
Israel.
A lot of places they don't connect the dots for undergraduates. It's just assuming they'll do that.
I love science, but it's an international component. It's not, there's no one
There
place
is a
where
pro and a
science
con
is.
because we move so much. You always lose and you always gain.
Right, yeah.
First, the lost words. All your environment, your friends, your workplace, and something new. But looking back, you've seen a lot, experienced a lot.
One of my very good friends who's actually coming today, she's a PhD student graduating at Loma Linda. She's from Egypt, and so she's like, hey, come over to visit, and we have another friend who's in Saudi Arabia who's a professor now. And so right now it's a little bit strange.
It's the worst city I've ever seen in all my travels. So much
It's
so
just
much
Yeah.
junk. The trash.
It's interesting. Yeah.
It's really, coming from Dubai, which is
Oh, right.
sparklingly modern,
Yeah.
the most modern place in the world right now. Cairo is just, but
I just want to do one thing.
things like
So
this.
timing wise for your talk, did you talk I know you
Yeah.
talked to Ali about that.
She said I have between 9 and 10:30. I saw
You have
what we should.
both sides.
I do lots of stuff.
Right.
Okay.
I can jump
There's,
and
there's ups and downs. Probably anyway, but yeah, that's good to know about Cairo. She's not in Cairo.
yeah.
We didn't make it as far south as Luxor. Everybody said, no, this is a place to go. Then you don't have to go.
Okay.
Yeah.
Okay.
And to see
Well, what we're gonna do? I'll introduce you. 9 o'clock. I'll introduce you.
But the
Okay.
Nile
You'll start about
River,
9:10.
all these bottles were floating there.
The poster session starts at 10:30. So what we want to
With
do
so
is
much
we'll go
history, you would hope that they
till
would have
about
some
10 o'clock.
Oh, I would love to see
And
that.
then we'll take some questions. We want to make sure we break in some time for questions.
Pyramids and
And then they'll have time right after that. So they'll be able to grab coffee, restroom break, and then go to their go to the posters after. So we have posters running on right now as well. And so our first, we actually broke our poster session into two. So you looked at a few. Okay. So I just want to make sure that we, just to give time for questions, that we go probably about 50 minutes or so with your talk. So 9 till about 10. And I'm going to see if Ryan can help me as well to field some questions. So we'll have a couple of wireless mics that we'll take around. And
A band,
I will
Yeah.
jump several slides.
Yeah,
because I put in
digital
slides which people might
projection
information.
of what
I think it's a lot.
you cannot take
It's
pictures
Yeah.
of
very easy to
in this one
Right.
area.
And we'll let you know when we get close to around 10 o'clock. We don't want to cut you off, but we want to make sure the students have time for questions. So.
These were live people 3,000 years ago.
Yes.
It's amazing.
Yeah. I mean, you know, as a scientist, I wonder if they look different, they act different, their genetics are different. You know, there's so many things
They were
that you
taller than we always thought they were.
Really?
Yeah, yeah.
Yeah, I would love to. I'd love to. We have just a few young kids right now, and so once they're a little older, we're keeping
Yeah,
them
when
running
our
around.
kids were young, my husband's were.
Yeah, I know when you mentioned that, it's exciting.
Yeah,
It's coming more.
that's
coming more.
Even though we-
Yeah, how is your grandson? He's in Cornell still.
Okay.
don't believe in that.
Yes. Okay. Yeah.
Colin.
Yeah, Colin, right. So he's not.
No, he's business.
Business.
Yeah.
Really,
Yeah, it's got some control of everything. So yeah, we're,
very unknown to us.
Yeah.
we'll just.
He'll go and make lots of money somewhere doing something.
That's what we want. Yeah, he wants to have his own
Yeah.
business.
This can be over.
No, he is now.
It works. Yeah. Yeah.
He lost some
good.
time somewhere
Well,
along the
we're
way.
thinking when you're here to.
No, he went from public school to private school and oh no, and then also Cornell, so he played some here. No, he's not finished yet.
Yeah, he's sometimes.
Yeah, but Ithaca became.
Hopefully some of these students will get the fire too. So
After Caltech, we worked for 4 years, for 10 years at Cornell.
where we're at here.
So
Yes,
we
yeah.
were at Cornell for 10 years, and then our daughter graduated from Cornell, and now the family.
Oh, it's a pleasure.
Can I ask one other thing of you?
Yes.
So, Dr. Salai's going to speak. I'll introduce it around 9 o'clock. He'll start about 9:10. We'll speak till about 10 o'clock, and then we need to field questions. Can you help me to field questions? Okay, so we have 2 wireless mics. So one will take this side, one will take that side.
Okay, yeah,
thank you, sir.
I'll walk around on this side then.
So you take that side, and then we just like kind of like.
I will show him the nice pastries that you
There
have.
we go.
Yes,
Please do. Please do.
excellent.
Please get something to drink, and we have a water bottle for him.
I got him one. I did get Phil one though.
I should.
So I'll go field this sign then. And,
Yeah, so right when he's done, we'll try to get it, if he goes much past like 10:05 or 10:10, we'll try to, because we want to get a few questions in at least, and then give students a little bit of time to transition over to the, because I have some more announcements at the end.
Okay.
So we'll probably have about 15 minutes for announcements, so 10 o'clock's about 10:15 or so.
Okay, so if you want to, if you would like to initiate maybe the questions and things,
Right.
I'll just kind of go over there and do
So
that.
right around 10 o'clock, I'm going to sit over here, I'll just stand up right around 10 o'clock,
I'll probably,
and then if you can sit wherever you want to.
I think my bag's back there, so I'll probably stay right there and I'll just walk up with it.
Okay.
All right, great,
Okay.
excellent.
Okay.
Anything else that I can do?
No, you're doing great, thank you for,
This is the one?
we always want to have somebody to just like attend
Yes.
to the, so.
Right, right.
Appreciate that.
Maybe after somebody was left alone. I would feel uncomfortable if I went to a
Well, I
place.
think sometimes you don't think of everything.
Right.
You know what I mean?
I did. It's a button shirt.
You have buttons on your shirt.
I have buttons on my shirt today. I did dress up.
Hey,
How's it going?
Good to see you.
Good, good.
Did you learn it from you, or you got
Taking notes.
Yeah,
Protege here.
just for the position you want?
yeah. I'm content, so that's why.
Yeah, right. I have a lot of growth now.
I don't want to be promoted. I'm good.
One day.
Go back to just teaching.
You got some folks.
That's what I'm trying to do.
It's Mark. Okay.
It's
I was in a couple of them. I
saved
don't
on my
know.
desktop. introducing him, and then we'll field questions after. Some students. Just quickly, more thank you. Very much appreciate your time.
No, delighted.
Thank you.
I know it's been a while.
And no Dr. Language this morning. Too early for Dr. Language.
They're being written together to remember.
Now, yeah, we can see.
All right, we're going to get started. If everyone can take your seats, please. Everyone, we're going to get started. If you can take your seats, please. Am I on? Everyone, we're going to get started. If you can take your seats, please. Just to let you know, we also do have some overflow seating. It doesn't look like we're going to need it right now, but we have 123 across the hall is open as well. So hopefully we can get everybody in this room, but we do have that overflow seating if you have others who are coming who aren't in here yet. We have plenty of seating up in the front here as well. I'd like to introduce myself. My name is Dr. Tom Furco. I'm an Associate Dean in our College of Arts and Sciences over our departments of Biological, Chemical, and Mathematical Sciences, our Division of Natural Mathematical Sciences, and I would like to welcome you today to our 17th Annual Natural Mathematical Sciences Research Symposium. As I was thinking about this, we've done this 17 years. This is probably the last year. Maybe we do have some this year, some students who are presenting who were not actually born the first time we did this. So we're getting to a point now where we've done this for almost 2 decades, and we are very thankful for all of our students who are here to present today. We're going to have 163 student presentations as part of our symposium. We're going to have 47 research posters and 34 oral presentations, and we also will have a keynote speaker here to start off our day. The
breakout of the day, just want to go over this. We're going to have our keynote address, again, starting in about 10 minutes or so. We had our first poster session today. I know it was very active from about 8 to 8:45. After this session, we're going to have another poster session that is going to start at about 10:30 and run till about 11:15. And that's going to be in classrooms on this floor and then also upstairs. So if you weren't able to attend the first poster session, so 3 classrooms, 103, 104, 106 around this side of the building, and then the patio, and then also rooms directly above, 203, 204, 202, 203, and 204. After that, we're going to go immediately into our oral presentations, which are going to be in 3 different rooms, or 4 different rooms, depending on which discipline you are in. So this will be from about 11:15 to 1 o'clock, and the biological sciences will be in this room here in Innovators. And then across, we're going to have chemical sciences in 123, math and physics in 124, and statistics in 125. So the 3 large classrooms just across. So we encourage you to stay for the entire day. I encourage you to engage with our student presenters, ask questions. So when we started this symposium, again, 17 years now, the goal was to give our students an experience to see what it's like to go to a professional conference. So we've modeled this after professional conferences. And if you go to a professional conference, you're going to experience the same types of format. You're going to have keynote address, you're going to have breakouts where you have posters, you'll have talks. And the entire goal of the day is for students to be able to present on the research they've done, but also for those who are considering research or students who are even presenting today to engage with other students, to build that research culture, build questions. So I encourage you, again, to ask questions, think about questions. We're actually going to have a question and answer time at the end of our keynote. So I encourage you also to think about some questions as our keynote speaker is speaking. So a few housekeeping details as we're going through. First of all, there is a program. Hopefully everybody was able to pick up a program. If you were not able to pick up a program, there is a code on the back that you can scan in order to get access to that. So we have both either digital or paper copies of those. If you're a student and you're here, which is a lot of you, we do have swipes for both before the keynote and also before each of the breakout sessions. So I encourage you to scan your ID card, especially if you're getting extra credit for any classes. That's how we're going to keep track of that. So we're not going to have sign-up sheets for any individual class. We're going to combine them all. So just make sure you swipe your ID before you go into one of the breakout sessions. The event is going to conclude at 1 o'clock, but that's not the end of the day. We have a separate event happening right immediately afterwards, which is a graduate fair that our biological sciences department has put together. So they have, I think, at least 10 graduate schools coming to campus today. So if you're at all interested in graduate school, professional schools, I encourage you to attend that as well. So you can go grab some lunch. It'll be for an hour, a couple hours after we're done here. So it'll be on the lawn out here by the engineering building. So again, I encourage you to attend that as well. And one other thing that we're going to do at the conclusion of our keynote address is, I think you guys got, students got an email from Ali yesterday who are presenting. Our speaker today, Dr. Zalai, has generously decided to donate his honorarium back to the division. And what we've decided to do is we randomly chose 10 students who are presenting today, and we're going to award each of you a $50 check just as an award for being here. So those are randomly drawn. I don't know who they were. Ali drew these with a random number generator. So 10 students who are here, we're going to just give you an award for presenting today. Okay. And that'll be at the conclusion of our keynote address. So now it's my pleasure to introduce our distinguished keynote speaker, Dr. Aladar Zalai. Dr. Zalai is here along with his wife, Margot, and also we have Dr. Hernandez here joining us as well, our Dean of our College of Arts and Sciences. Dr. Zalai is internationally recognized for some groundbreaking research utilizing live microorganisms, such as bacteria and viruses, both to diagnose and treat human cancers. His work has led to over, I want you guys to just grasp the numbers here, his work led to over 350 peer-reviewed scientific papers, 170 patent applications, and 90 issued patents. That's a tremendous amount of work, and that's what a life dedicated to research will generate. Among his numerous accolades, Dr. Zalai has been awarded the Wilhelm Exner Award for Excellence in Science from Austria, and the Alexander von Humboldt Prize from Germany. And I'm just going to list a few of the universities he's associated with and companies he started. He's associated with the University of Würzburg in Germany, University of California, San Diego, and Stanford University, both here in California, and also 3 companies that he has started. Genelux Corporation, STEM Immune Incorporated, and Immunolux International. I believe I got those right, hopefully. Dr. Zalai, again, is going to address us today. His talk is going to be on the smallpox vaccine, a future cancer vaccine. So join me in welcoming Dr. Alagdar Zalai.
Should I stay down here or should I come up there? Good morning, everyone. One could not have a more precious morning than seeing 300 graduate students and faculty and thirsty for science in an innovation building. I did not know your university. You know, I'm coming from research universities. But I am very delighted that now you arrived with your president and with Dean Hernandez to a stage where you convert now your university from a teaching university to a research university, which also will lead in funding and also perhaps even industrial input, which you start here companies. So with this very short introduction, I would like to, I put in a large amount of slides because I did not know what questions will come up, but I would like to go from the start to the end. Before I left Cornell as a faculty member, I started to work on light-emitting organisms. And we cloned the light-emitting genes, and that time it was no GFP, no Ranilla, nothing that time, and we went on with everything. Finally, our research ended up with light-emitting bacteria, which emitted even 2 wavelengths of light. One was luciferase, which was 268, and the other one was GFP. And this light-emitting proteins we then injected into mice and with bacteria and tried to see where the bacteria go. And with Siemens, that time I constructed an imaging equipment, and this imaging equipment could monitor the live pathogen movement inside of a living organism. See future, can we do it in humans? So just before I go in, the title of my lecture is here. My affiliations are here. I have to make some disclosures. You are also in public meetings obligated to make disclosures. So I founded Genelux. I started, as Dean Ferko said, STEM Immune, and also I am now also president and CEO in addition to my academic title of Immunolux. This is a slide. If you have a mouse and you put on a human tumor, just say it's a brain glioma, you see an implanted tumor, if you talk about. This implanted tumor, then when you inject this mouse with light-emitting bacteria, lo and behold, we found, and it was a very, very first publication in that direction, Nature were holding it for 6 months because they did not believe me. And we had to do over and over prove it that the pathogens, when you inject it into live organisms, they find the tumor, go into the tumor, multiply in the tumor, and based on light emission can be monitored as diagnostic. So we discovered the targeting of tumors as well as diagnosis of tumors. Then we asked, fine, bacteria live in the tumor, but what do they do to the tumor? Why don't we take a pathogen which is killing the cells can replicate only like a virus. And I decided, among major oppositions, that we should work with vaccinia virus. When they asked me why vaccinia, because we, at least the senior one among us, still have the patch from the vaccination against smallpox. And this vaccine is a cow virus, cowpox virus, was used in humans since 1794. It is as old as United States, and hundreds of millions of children were saved from pox. And this is why I took. All the clinical trials were done, even by children, and even the pox vaccination. And this virus, we made also light emitting. And lo and behold, when we went onto the light emitting virus, then eliminated the tumors the same way like bacteria did. I just wanted to show you this slide because it is a historic slide. This was when first, where am I? I cannot see it somehow. Then it was, sorry, I just pushed the wrong button. So this virus was injected, as I said, 1796 in London into a boy, his name was Phipps, who had pox. The Jenner, a country doctor outside of London, recognized that the milkmaid who milks the cows never got pox. And Jenner took from one of the milkmaids the serum and injected Phipps, and Phipps got treated. So this is the history of vaccinia virus, and now we are using it in cancer therapy. So we introduced our light-emitting proteins into this virus. I just have to go a little bit faster. And now it is expressing photon emission as well as fluorescence. You have the double label now. It is a common news. I mean, please, it was 22, 25 years ago, yeah? And we put it into ovarian cancer cells, and we said that light-emitting genes did not interfere with the virus at all, functions perfectly well. And then just very recently in Würzburg, we crystallized RNA polymerase of the virus and obtained the crystal structure. And we discovered one very important thing, which is a total surprise and we do not understand. This virus has in the RNA polymerase a tRNA incorporated. And this tRNA is glutamine tRNA, and the virus can replicate happily only in cells which are in abundance of glutamine. So somehow somebody there or nature worked it out that the virus polymerase sensing the glutamine and the replication is go. Now, when we introduced this glioblastoma tumorous mice with the light-emitting virus, you could see that again, similar to bacteria, only the tumor lit up and nothing else. And when you looked at the GFP, you just can see that the pockets, as a tumor grows, only the human tumor, not the mouse tissues, the arteries and so on, are infected. Only the tumor is. So just look at now all our chemical companies and everyone who is injecting chemistry, whatever. Number one, this chemistry is not specific for the tumor cells only. the tumor cells and other cells are also killed. Number two, these chemicals do not find specifically the tumor like our virus finds the tumor. And number three, if I show it in the next slide, the virus is also killing the tumor. You can see it very clearly how the lights goes away, and it doesn't matter if you take femoral vein injection, tail vein injection, or intratumoral injection. The tumor is being killed. So the route is not important, which all our chemical pharmaceutical companies are trying badly to achieve, which is impossible. So the next slide shows you. Now I am in trouble. Oh. The next slide shows you that the distribution of this virus, because the virus has this God-given capability, we can plate the virus and count the infected foci. So if we can even quantitatively determine how many virus particles have to be there to kill a tumor or kill a tumor cell. So it is not just like with chemical, and I'm not trying to bash the chemical industry, please. It is not my mission. The mission is to take out the advantage of this virus so that it is quantitative, finds a tumor, and also locate. You can see it that if you look at the tumor here, 10 to the 9 particles per gram tumor, and if you look at other tissues, just what the blood is carrying, at least millionfold or much less. Yeah. So then we started to do different tumors and different cancers, and we can see that not only glioblastoma, not only cervical cancer, but also mesothelioma, lung cancer, is being killed. And this slide just summarizes very quickly what I tried to say up to now. Though the virus is coming into the tumor, infected the tumor cells, killing the tumor cells, releasing the tumor cell protein, and practically immunizing against cancer, and invites cellular immunity. They also, now I just talked about solid tumors up to now. We also looked at metastasis. This virus finds all the metastases everywhere. You can see it here, and also even on lung surface, metastasis, micrometastasis, and so on. And also the circulating tumor cells. As you know, if you have a solid tumor, the solid tumor spins off metastasis on different sides, and when the metastasis is too much, you will have elevated circulating tumor cells, which is killing the patient. And it was also very important that you take a drop of blood and add the virus, and you could see how many circulating tumor cells we have. So this was all done. I just wanted to tell you how we went from light now to individual tumor cells floating in the patient body. There are the circulating tumor cells, which we also labeled. And we went also to leukemia, and we can take a patient leukemia blood, and you can see it very clearly that the aggressive leukemia blood also is very clearly identified with the virus. It doesn't seem to work, my pointer. And here also these aggressive diseases, you can see again from directly patient's blood what is happening, and we can use it for solid tumors as well as liquid cancers. Now, we said not only light can be put on this virus, genetically engineered, we can put on any other gene. Can we deliver with the same vaccine here into the tumor other genes which we can express, and the tumor can be functioning as a pharmaceutical factory inside of the patient? If vaccine carries that, and that, and express it in a very high level in the tumor, can we produce it? And this was also very, very successful. we took certain genes and, for example, the anti-VEGF antibody from Roche and put it onto the virus, and we have a fantastic tumor reduction when we have the Avastin here from Roche, and if you have the Avastin and the virus is here, the tumor grows, so completely inhibiting. So single chain antibodies are one of the targets which we put on the virus. So we are not using now light-emitting virus only, we are using added expression cassettes and producing it in the human tumor as fermenter, as a pharmaceutical factory. Just see that little pieces of Roche we are putting into the patient's tumor. We also added diagnostic capability. We added the sodium iodine symporter and the human sodium iodine symporter, and this virus, when we inject it, takes up iodine, and you can use the certain iodine detection technologies very well worked out in medicine also here. Oh, God. Something is wrong with this. We have to exchange it. Yeah. So I move it, and now once more. Now. Yeah. And you can see it now that iodine expressing virus is there. The next one. It's not working. Now could you just push them if possible? Yes, and also this iodine uptake expressing virus, when we used it to treat triple negative breast cancer. Thank you. I know.
That one doesn't have a laser on it. This has the laser. That doesn't have the laser on it.
I know. Thanks. The triple negative breast cancer is very, very difficult to treat. And this virus now, with iodine uptake, can treat also triple negative breast cancer in mice, and it is also part of the future clinical trial. It was done together with Sloan Kettering. So I hope now, yes, now we are going into now perhaps human clinical trials. I talked to you up to now mostly lab data, cell data, mouse data, and human sample data, but now we are going in with a virus which is GMP produced. It's also FDA approval, and everything is necessary. There's lots of work, lots of money goes in, but we have now certain virus type GMP produced, and we can use it and put it into patients. Now, we started out and work on different clinical trials, Sloan Kettering, UCSD, University of Tübingen in Germany, Royal Marsden in London, and in Surrey, and we did clinical trials. Perhaps the details are not important. I'm showing it to you not to brag or try to impress you. I'm showing it to you what it takes to go from the lab to the GMP manufacturing, and then go to different clinics. So at every clinic, you have to deliver a clinical trial protocol. This clinical trial protocol has to describe every little detail, how many plaque-forming units or virus particles you are putting into the patient, how long and how do you harvest, how do you prevent cross-contamination. Just imagine, at FDA, we have administrators which are very, very important. It took me 6 months to convince the FDA that the human patient injected with vaccinia will not spread the vaccinia to husband, wife, and neighbors. And how do I know that? Because I took bath when we were little with my brothers, and one of us or two of us had the vaccinia infection, and in a bathtub and everything, and none of us ever got infected with vaccinia, and how many million people. But again, the shedding and cross-contamination in the today's FDA took 6 months and enormous amount of activities which we had to do. But so we started, and it was a long time ago now. We injected that time over close to 300 patients here, and these patients were tolerating everything very well. In Germany, it was very, very important. The ovarian peritoneal cancer, with 10 liters ascites in a lady's abdomen, after 4 days, not one virus particle was found in the ascites, which was practically full before. So in 4 days, the virus took out from 10-liter liquid the viral, the tumor cells completely. So again, it worked in this cancer. We also isolated the virus from live patients, and you can see the thing. Also tumor biopsies we took from intrapleural administrations, and also the patients were also colorectal patients, and also the virus could be. And the Verco, I mentioned to you that this is one of my most beloved pictures. It was the very first time in London that we could show a patient growing with a little metastasis on the chest in the hospital bed. I sent from San Diego an imager with a big gooseneck, and the patient was scanned in the dark, and it was done, 2013. And I called up my friend in the Royal Marsden. I said, "Sir, now I have succeeded to label the British." And he said, "Yes, sir." But I think it is a major achievement. When I went to a conference, I was yelled at by Americans that, "How did you ever get the permit to put in GFP into patient?" I said, "I did not put in GFP into patient. We put in vaccinia virus expressing in patient." And the United States GFP was not allowed to put into patients. Yeah. All right, so why don't we continue and just say that we have key consideration. Multiple dose could be used. The optimal dose was roughly 10 to the 9 particles, no dose limiting toxicity, and nothing plateaued. So then we moved on. And I just wanted to give quickly thank you to all the people, from students to doctors, and most importantly to our patient, to this portion of the lecture. Now, the company, Generax Corporation, started to take the virus alone and inject patient, and we are now, I am humble but very proud to say, at NASDAQ, and in phase 3 trials, platinum-resistant ovarian trials, where every woman after reoccurrence 6 months later is dead, and the virus is saving the patients. We are now in phase 3 trials in Genelax with virus alone, and it is now expedited. So I hope by end of this year we will be approved and we can save human lives. Just imagine what a privilege to an immigrant to come to this country and try to help half of the world population. I do not want to go through with you the different details. Important is that that we are doing the ovarian cancer as well as we are doing lung cancer, and again it's phase 3 program. The company funding is very, very important because these clinical trials are enormously expensive. I mean, it is very, very rare that a trial with 350 patients comes out below $5 million. And this $5 million, the president of the company who started this have to come up with. Before that, FDA do not even talk to you. So it is not that way that the federal government very generous and say, okay, if you do this important clinical trials for our women population, we give you that. No, you have to bring the money, and when you have everything, we can start. Yeah. All right. So next slide is again the lung. And in the meantime, one of the naive graduate students took the virus and took mesenchymal stem cells, adipose tissue derived mesenchymal stem cells. And lo and behold, we saw that the virus infecting the mesenchymal stem cells and multiplies mesenchymal stem cells. My thought was right away, in that case, we could take the patient's own mesenchymal stem cells, put the virus in, and put back the virus, the mesenchymal stem cell carrying the virus into the patient. So it is like an autologous cell delivery system to protect the virus from our degradation or defense. And this was the very, very important discovery. It was discovered by a graduate student turning postdoc. Now he's at Stanford, Ivan Petrov, and we started this direction. But not only mesenchymal stem cells. It turned out that pluripotent stem cells all were infected with the virus. So practically, even organ-specific autologous stem cells could be used as carrier for the virus. And this is now, as you know, at NIH as well as everywhere, a huge fight between autologous stem cell as carrier of the oncolytic virus and the allogeneic, the non-homologous system, and on. And I am a strong believer of the autologous system. The patient's own cells should be taken and put it back to the patient. But this is not kosher with the pharma industry, because the pharma industry would like to make 5 million vials from one cell line and sell it to everyone. Doctor-driven autologous system, the doctor takes off your cells, separates the stem cells, puts a virus on it, and 2 hours later injects a patient. So I try to support the personalized medicine much, much more. So here we did it in Palm Springs as well as in Beverly Hills. We took liposuctions from patients, cancer patients, and from this liposuction, with a certain machine, the stem cells can be isolated from fat cells and grown for certain hours. And after that, we call it adipose stromal vascular fraction with a stem cell fraction, and then mixed it with a virus, and the virus infects it beautifully, and then putting it back to patients. But before we put it back, we also discovered that this mixture is preventing the serum to kill the virus. So it is even protective to the virus enormously. So not only autologous, but also taking the virus and protecting it against a host. And this is perhaps the most important slide, and I do not want to dwell on it any longer. If you take an intratumoral injection, you can see that whether you take cells alone, cells plus virus, or virus alone, the tumor regression is the same. But if you take IV injection, you inject it into the blood, that how the cells protect the virus from the inactivating ability of the patient. So it is very, very important that we protect the virus. I do not want to be political, but perhaps I have to be a little bit. If you look at this red box here, this is our American smallpox virus. All nations have different pox viruses, and all nations declared it as their own. US has one, China has one, Brazil has one, a large nation, Europe has one. They are all vaccinia virus, just a little bit different isolates. But it became now a political tool, because if you would like to take this virus in London, they will not inject you because you are not British. If you try to get the virus in America and you are not American, you will not get the ACAM 2000. So now, but for tumor purpose, now really every nation has a stockpile which is stored by the US military, the German military, the British military, as safety waiting for biological terrorism against pox virus. Huge containers. But practically, our drug for United States sits somewhere controlled by Washington. I know who the people are because I got it from them. And we have enough stockpile in United States to cure every cancer with vaccinia. So the values, which I'll show you later, where one patient treatment costs over $500,000, could be reduced to less than $50 because we have the material, patient has their own fat, and you could take it and mix it. So what I'm saying is it's a huge economical battle, political battle, but if our stockpiles are released for cancer treatment, we have at least a huge chance to cure cancer, or a lot of cancer. When we go now further and look at the second company which I started, STEM Immune, this is now using immune cells and virus. So the stem cell then virus. And this company, I just do not want to bother you with it, is it now analyzing what is happening to the virus in patients injected, and also the memory stem cells which are and they are enormously enhanced. I lose my concentration on this pointer, so I don't know which one to put. The virus, so central memory stem cells, so they are enhanced in patients' blood after treatment. Most importantly, we used vaccinia virus and stem cells in this thyroid cancer patient. He was a stage 4 patient, rejected, go home, die in peace. We injected once with his own adipose tissue-derived stem cells plus our vaccinia virus, and you can see that after 85 days, everything is gone. Now he is alive 7 years, it is an 8th year after. He just wrote me yesterday an email, and every time the year comes by, he's thanking me. How many young scientists, like I used to be, have the chance to show other young scientists today, 30 years later, that this is what my and my associates' research does to patients? I'll show you the next slide, which is even much more drastic. It was a head and neck cancer, and after 190 days, all this terrible cancer. This patient was also stage 4. He could not even sleep anymore. One injection, his own stem cells and vaccinia virus. Now, again, we are very thankful, very humble. The people are Dr. Feller in Palm Springs, Dr. Landa in Palm Springs, and one of our colleagues, he died in Beverly Hills. He was also a plastic surgeon. So it works. FDA came in between when we went to phase 2 trials and asked, can you tell us precisely, if you have 5 patients, take from each fat, how many cell types and what is the quantity of the cell types of this, because otherwise we will not allow you, because you have a stem cell fraction, but you do not know how many mesenchymal, how many that and that and that. And they did not allow to go on with phase 2 because we could not give them the precise number. The estimated 10%, 20% was not enough. And I think it is absolutely unbelievable that patients' life can be saved. The number of their fat contained stem cell is a determining factor because we do not have a technology for it. So I just go on and not politicizing. We were very lucky. We treated 24 patients and 2 leukemia patients. In a phase 1 trial, you can follow them up to 4 to 6 months. I cannot go after and ask after 10 years now or 8 years now, how are you? But they can come to me. We have, for example, in Incline Village and in Cabo San Lucas, a gentleman, he is 95 years old. We treated him 10 years ago with stage 4 prostate cancer. And he is alive and everything is fine. So we have at least 6 or 7 people who are communicating with me. They are thanking me every year that I am still alive and thank you. So what a humble privilege to be part of it, and we have to go on, and we need an administration in Washington who supports human life and not supports some kind of idiotic questions. The efficacy with our own stock in Washington, ACAM 2000 is ours. Yeah, just to see, we have a beautiful system. We had a beautiful vaccination system for vaccinia. One doctor from Riverside can write to CDC and say, my patient is going to Africa. There is a pox infection. Could you please send me a vial? CDC sends the doctor the vial injects the patients take off to Africa. This injection is possible. Against cancer, it is not possible. I was told many times by very high-level FDA officials, Professor Salai, you still did not realize that cancer patients are not healthy children whom we are vaccinating. I said, I do realize, but we saved from the 25 what we injected at least 20 up to now 7 or 8 years. Why we cannot continue? Yeah. So it is undecided. I think I said enough. I just would like to move on. Now STEM Immune converted to Khalidi when it went public, and now they are doing clinical trials in malignant glioblastoma. I do not want to detail on it, together with City of Hope and Case Western, and these experiments are going on. And thanks for all these people. Now, just, I don't know how much time do I have?
About 15 minutes. 15.
Oh, it's perfect. When I was president and CEO beside my academic position at Genelux, 2014, I stepped down and I started STEM Immune. 2021, I stepped down and I started Immunolox. And Immunolox is now an existing small company filing mostly patents and for my graduate students data which we are obtaining. Immunolox is using immune cells as carriers for vaccinia. Lo and behold, we discovered that when we injected T cells, the T cells are taking up the virus, and the virus survives longer in patients' own T cells. Now, when you see here, this is a neuroblastoma cancer cell line from children. Then, if you add, if you have only the CAR T ratio 2 to 1, it is very tough to kill it. But if you have CAR Ts and infected with vaccinia, you can see that this killing the tumors very efficiently. So, and we can see it also in 3D cultures and vaccinia infected CAR Ts are much better than CAR Ts alone or vaccinia alone. So somehow, someone up there designed that if the immune cells are not enough, vaccinia will teach you how to be better. And that time I did not discover it, did not take it that seriously. I said, oh, something accidental. But when we went on and we did all these experiments in a most experimental fashion, not in humans up to now, and you can see that the autologous CAR Ts, allogeneic CAR Ts, we can subdivide the engineered CAR T system beautifully, and vaccinia is enhancing the CAR T activity in killing tumors. I think this is enough from that. I just wanted to add one more thing. This is a technology, the CAR T technology, when one patient pays over $500,000 to make his or her own CAR T. CAR T stands for cancer receptor. So the cancer receptor is put into the T cell genetically. The cells are grown up 4 months, and after that it can go back to the patient. So in fact, it is an autologous T cell mediated tumor therapy. So again, we can enhance CAR Ts normally for liquid cancers, but hopefully we can also broaden it to solid tumors. Again, the CAR T and T cells as carriers, and you can see that you see T cell and tumor cells and CAR T cell and tumor cells here in comparison, this side and this side. But when you go on and do the different infections, you can see that the CAR T is enormously, in conclusion, the CAR T cells can be infected with vaccinia, not toxic, and the CAR T significantly impairs survivability, but CAR T and vaccinia is enhancing tumor killing. Now, the last chapter of our companies is using the brain immune cells. As you know, our brain has enormous amount of microglia. The broken down proteins, the pathogens and everything are eliminated by microglia. It was again one of my wild aberrations. My student used to say, I told them that why don't we try microglia? Who knows, perhaps they will do the same thing. And lo and behold, we did. And again, the BV2 is on microglial cell lines, and microglia is killing tumors. But if you look now, this infected microglia here, I don't know how well you can see it from the distance. Here, they are much, much better than microglia alone. They are all also cell lines from neuroblastoma from children cancer. My laboratory is now, I forgot to say, the most important thing is focusing only on children's cancers. You know why? Because from all our clinical protocols when we submit, we have to write exclusion children below 12. If you remember, our smallpox vaccination were all done on children below 3 or 2 to 3. How logical FDA is. Cancer treatment is below 12 with vaccine prohibited. But pox protection with the same vaccine, this is our smallpox vaccine, is not. So again, I am working now only on children's cancer and try to help the most unprotected one and also lifelong lives with children. So we did also the 3D cultures, and it works beautifully in 3D. You can see here, I do not want to be detailed, you can see here virus alone, it's just the glial cell alone, and then organoids we did, and you can see that here that the virus and the infected glial cells are killing the tumors most efficiently, 10 times to 15 times better than glial cells alone or virus alone. So now I do not know, is our projecting people, could I please have the last slide, not the last slide, the fourth slide with Jenner, the little guy who got infected. Yeah, if we can do it, if not, it's fine. No, the very first or very second, third slide, yeah? The colored slide with a child. So, I hope it was not too confusing, but I took you through the simple tumor finding with self-glowing systems. This self-glowing system was put on vaccinia. Yes, thank you. Just keep it on, please. Thank you. Put on vaccinia. Vaccinia does the same thing with bacteria, but kills tumor cells by infecting them, oncolyzing them, and on. And now we also discovered that vaccinia is creating immune stimulation. The cancer cells are live. The cancer-specific proteins are practically vaccinating and helping the body to cure cancer. Now later, the cancer cell, the virus delivery could be made much more efficient by protecting it from the host's own immune cells and giving it to adipose tissue-derived stem cells. And now, in the very late phase, I can proudly say that perhaps it's a brand new discovery in cancer research that immune cells, T cells, CAR T cells, as well as microglia are improved by vaccinia infection and killing cancer cells much more efficient than each of them alone. So with that, I would like to thank you for your patient audience. Sorry for this pointer business. And I would like to stop here and take any question. And thank you that you young people listen to something which is perhaps far away from your daily math or chemistry lectures. Thank you.
All right, let's give a round of applause. We're going to have about 15 minutes or so for questions. So if you have questions, we ask you to raise a hand. I'll have a mic on one side. Dr. Fuller will have a mic on the other side. I'm going to start off with one. I'm a chemist, not a biologist, but From what you said initially, is it possible in the future that someone can get an injection of the vaccine and it will prevent future cancers? So immunize someone against cancers that they don't even know they would have in the future.
It is a very, very important question. As you know, the senior ones, we are vaccinated and we are protected. Now, just recently a paper came out in Science where it shows that women above 50 getting now more cancer than before the vaccination time. Just remember, 1978, the obligatory smallpox vaccination was stopped. We eliminated smallpox. Now, 50 years later, you see that I do not know that it has anything to do with prevention by vaccinia virus, but practically we are trying to save on smallpox vaccination and eventually do not eliminate the occurring cancer at early stage. Definitely, I would suggest that vaccination against cancer with smallpox should come back. Yeah.
Questions, please raise a hand and we'll bring the mics to you.
Yeah.
Yeah, it seems for tumor cells, right? It's constantly replicating. So it's not surprising that viruses would infect them because they provide a way to replicate the virus ad nauseum. So is it possible other viruses can do the same thing?
It's very, very important. We just wrote a review in Frontiers of Immunology about the oncolytic virus therapy meeting in Holland last November. And there are at least 8 to 10 different viruses, adeno, herpes, and odd, was being used. All the virologists, when they discovered what we discovered 25 years ago and published, that the virus is killing cancer, all the virologists converted to oncolytic virus therapy. Yes, we have several other virus strains which are even SVF is even in glioblastoma cancers in clinical trials. Yes.
And just one more. What's so special about the vaccinia as far as killing those tumor cells? Do you know the mechanism?
So it is very important. Vaccinia, most of the viruses have a cell surface receptor where they get attached and get engulfed and so on. Vaccinia does not need any cell surface receptors. Vaccinia is somehow engulfed by the tumor cells, and then once it is inside, carries everything on the vaccinia genome. I did not say anything about vaccinia virus has 200 transcripts roughly, or 200 proteins, and everything what is necessary for the replication is encoded in the viral genome. So you do not need to add, and you do not need anything from the host, the nucleus, anything except amino acids.
All right, we're going to take one from Dr. Flores.
Over here. Great talk. My question is regarding the mechanism as well. You showed this remarkable phenomenon at the beginning where the virus is attracted to the tumor site. So what is the mechanism behind it? What is attracting the virus to the tumor site? Is it the abundance of immune cells, the tumor secretions? What is it that is attracting it there?
The question was, I do not know if anybody, what is special about the tumor that vaccinia can go into the tumor and replicate? The true answer is nobody knows. But the fact is that when vaccinia is engulfed by the tumor cells, then somehow an enormous replication occurs. And this replication lyses one cell and then the next cell, and the next cell, then you have an entire focus of infected area and attraction of the immune cells. But the entry is not clear.
Again, if you have a question, just raise a hand and we'll bring the mics to you. We can get a couple questions at one time.
Yes?
Just give us a second to bring the mics.
Hello. So you said that this virus doesn't really need a specific cell surface receptor to attack the tumor. So how is it specific to cancer if it doesn't really have something it's recognizing to get into the tumor cell?
So once more, specific?
Specific to the tumor. So why does it not lyse other cells within the body?
Yes. The other cells cannot take it up before our immune system gobbles up everything as a foreign. The uptake is here somehow, again, is not very well known, but the uptake is tumor cell specific, and the capability of the cells, of the virus, to replicate in the glutamine-rich cell environment, which are the tumor cells. So it is somehow like a planet. They're communicating with each other, but we do not know what is the reason why the virus takes over the entire tumor cell.
Okay, thank you.
Thank you.
I think we had a question up here.
I just wanted to know, so with the administration of the vaccine, are patients also given chemotherapy alongside it?
This is a very good point. I did not say, perhaps you saw it on the text. Virus alone is perfect, but to satisfy FDA and not to lose the pharmaceutical companies' research support, we did also lots of combination trials. And yes, for example, the platinum cisplatin combined with virus is better than cisplatin, much better than cisplatin alone, and also much better than, not much, but somewhat better than virus. Yes, chemotherapy is not neglected, and also I think enormous amount of clinical trial work went into the chemotherapy phase 1, phase 2, phase 3 trial, and this data can be used now in combination with virus. It's a very good point.
Any more questions?
Thank you. That was a great talk. I'm wondering for GBM patients, can the oncolytic vaccinia virus be effective if the patient doesn't have a compromised blood-brain barrier?
I did not understand the first portion.
So for glioblastoma patients, like patients with brain tumor, would the oncolytic vaccinia virus reach the cancer? Like, would it be able to bypass the blood-brain barrier?
So, if I understand correctly, we did even clinical trials in pre-op and post-op patients. So it was glioblastoma patient, and try to see whether you, I hope this was your question, whether you removed everything, still something stayed there, or where is a virus going. I think the majority of the virus is eliminated. We hardly can find anything in the solid tumors. Of course, distributed micrometastasis in patients is very, very difficult to say. Was it the question or not?
The question was, does the virus pass the blood-brain barrier?
Oh, this is partially answered it because we did the glioblastoma. No, the virus is not passing a blood-brain barrier, but we do not know if we do not take biopsies. What we know is as I showed you in the clinical trial, we are doing real injection into the head of the patient and into the brain. But we systemically delivered, or perhaps I should correct it a little bit, a systemically delivered vaccinia virus into a glioblastoma patient. It was a stage 4 glioblastoma patient from one of our major clinics. And the patient, after 4 months, it was a stage 4 patient, started to use his bicycle up in Cleveland. He was from Cleveland Clinic and fell off the bicycle without helmet and died. But practically this was one patient where we systemically delivered large amount of base into the glioblastoma patient. The patient was practically cured.
Yeah, we're going to go up in the front and we'll go do one back here.
Real quick, so in the case of widespread metastasis for a patient, yeah, sorry. So in the case for widespread metastasis in a patient, would one injection where it targets the glutamine-rich areas be enough, or would it require multiple injections?
It's a very important point I did not have time to go into. In the phase 1 trial, we even did dose escalation and multiple injections. We find that the multiple injections will not eliminate the virus earlier, just create immunity again, so multiple injection can be made, yes. And we did not find either dose-limiting toxicity. We went up to 10 to the 11.
So when you administer vaccinia virus into patients, do patients get immune response, or do they undergo inflammation?
This is a very good point. I partially answered it, I think. Yes, the patients have very little immune response. We did these experiments in London just to see that if you inject and 2 weeks later you inject another one, will the virus be eliminated? No, somehow, miraculously, the immune response is not increasing.
All right, additional questions? Yeah, I'll come over. Again, just raise your hands and we'll stage the mics with you.
So I know you talked about the difference between autologous treatment and allogeneic treatment, mainly because autologous treatment, the receptor and the donor are the same person. Would you be more open to allogeneic treatment because of, for the sake of accessibility?
So this is a very, very hot issue in our highest academic levels. Why I tend to support autologous is because it is like blood transfusion or infusion. If you take your own blood and put it back, you can do it in 2 minutes, and no investigation is necessary for pathogenicity or anything. If you get something from someone else, it is much more proof is necessary. Now, we published a paper by which we add a certain stem cell fraction to allogeneic blood, and lo and behold, it will become like autologous. So one can control the allogeneicity with the patient's own stem cells.
All right, we're going to take one last question in the back.
So I know you had said that the immune response isn't really affected, but what about for people with autoimmune diseases or immune deficiencies? Has there been an immune response to that?
Can you repeat, or can someone repeat? I did not understand everything.
Here. When you're talking about people that have immune deficiencies or perhaps a compromised immune system,
yes.
What would this do to them, maybe?
Yes. So, thank you. We did not distinguish, like you do it with vaccination among healthy people, immune deficient and immune compromised. Most of the cancer patients, and especially the stage 4, are immune compromised patients.
All right, let's thank our speaker again. And Dr. Sly is going to be with us the rest of this morning with the poster session, and I believe with the oral presentations as well. So please engage with him. If you have other questions, please catch him and ask. That's why we're here. So thank you, sir.
Thank you.
All right, we're going to do a couple more things before we break, so stay where you're at. We have some money for a few of you, so you probably want that. And I want to acknowledge a few people. Let's acknowledge the people first, because I know you all are going to, once you get the money, then you're going to probably want to go and do something. I want to acknowledge those who have helped out today. So first of all, our Career Center, Cross, they hosted our morning snacks, so thank them. We are going to have refreshments after the talk here as well, so make sure you grab something. And also, again, the Career Center is always open for resume interview help, taking headshots for social media posts for promoting your work, so make sure you take advantage of that. We had many NMS faculty who mentored and guided students throughout this season for the research presentations to be able to be done today. So most of our faculty in NMS aided students in some way. I do want to acknowledge those, though, who reviewed abstracts and also those who are moderating the different sessions today. So if these faculty could stand up. So first of all, Dr. Annabelle Flores for Biological Sciences. Dr. Andrew Zhou for Chemical Sciences. I don't know if Dr. Zhou is here. He's not feeling well recently. Dr. Robbie Willett for Math and Physics. Dr. Esther Lee for Statistics. Those were our reviewers. So whenever you submitted your work, they went through and made sure that it looked like it was a viable project and something that should be presented at the symposium. We also are going to have moderators for the oral presentation. So Dr. Ryan Fuller for Biological Sciences. Dr. Jen Nalbandian for Chemical Sciences. Dr. Mike Sill for Math and Physics, and Dr. Esther Lee again for Statistics. So again, if you are presenting during the oral presentations and you haven't connected with them, they don't have your talk, please make sure that gets loaded onto the computer before you go to see the posters. For the posters, we also have poster hosts. The first time we've done this, we had so many posters this year that we had a few faculty who were helping out with posters. So Dr. Efat Zaidan Hartoum for Biological Sciences, and Dr. Satoru Suzuki for Chemical Sciences. We had a few student workers helping us out this morning. Natalie Tanaka, Jade Maldonado, Annika Cardinal, Silas Segal, and Annabelle Bruzas were helping with many factors this morning. Also, I want to acknowledge Dr. Fuller for helping with the liaison with Dr. Zille. And finally, a few folks who really made this happen. I think most of them are standing in the back corner there. Laura Flood, an NMS Administrative Secretary for Biological Sciences. Rebecca Montoya, our NMS Lab Purchasing Specialist, came in today as well to help. Jamie Corbett, who's our new NMS Administrative Secretary for Math and Physics. And finally, the person who really put this all together, I think it's probably taken a year off of her life working this past year, so she may need some of this vaccine, given how much that she's been working and doing overtime for this. Allie Floyd, our NMS Director of Academic Operations. So finally, before we break for the posters, and again, after this session, we're going to have some refreshments, but I'd encourage you to go around to the posters. Posters are going to be on the first floor and the wing over here, and then upstairs as well. I do want to give out some cash awards. I originally thought they were checks, but this is cold hard cash. So students who don't know what to do with cash, maybe give it to your parents and maybe they can deposit in your bank account. Then you can use Venmo and Zelle and other things to actually use it. But this is a piece of paper that's actually worth something to you. So if you don't know what to do with that. So I'm going to call out these 10 names. Again, these were randomly drawn. There was Ali did this, so there was no faculty input. None of us had any influence on this. So come on up. I'm going to hand it to you, and we're just going to have you guys stay up in the front here as I hand these out. So Kimberly Alva, come on up. Sharon Azariah, come on up. Nathan Bang. Is Nathan? Okay, Nathan's coming up. Xander DeWitt, come on up. Elizabeth Friederang. Is Xander here? Is Elizabeth here? All right, I have $100 here that I can do something with later then. Gracie Iverson. Gracie, come on up. Dustin Lucero. Dario Rodriguez Rueda. And two more. There's still a chance. Tristan Ryan. And finally, Javier Tornero. All right, and again, these are randomly chosen, but they do have $50 that maybe you could go to lunch with them today. So again, thank you all for coming today. Again, don't leave now. Make sure you go to the posters, come back to the talks. If you are going to the talks, again, for everyone, make sure you do get to your talks. And when the talks start, 11:15, we're going to start right on time so we can finish by 1 o'clock. Thank you again for coming.
They work together. did all these things, but he was involved in nothing so much. So they're very close. And they worked on the original imagery together. No, he's still involved. He doesn't hold official positions. Or I didn't. Yeah, so we excellent. Wonderful. Can I introduce the son of Matt? Yes, it's just right around the hallway. We can go look inside. Absolutely. Yeah. That's all right. What do you know when you're presenting? Is it just I didn't know you did. I was just trying to. Could we maybe start with that? So then you can start and then go over that. Would that be better? Okay. Let's try to that. Okay, I'll make those changes for you guys. Okay. Yeah, yeah. Of course, yeah. So start here. We're the best. And then you can go to things. Yeah. All right, good. Looking forward to it, guys. Take care.
Yes, it is killing more, but it is prohibited. You cannot take 10 different viruses, box viruses, not cow pox, and 10 different box, and makes a super virus. I mean, it's like biological warfare on the first scale. They did it in City of Hope. I did not want to talk with people calling me up, just that it's a prohibited virus. You cannot take box viruses from cowpox, monkeypox, human pox, chopped up, ligate them back together and see who is the best one to kill human cells. We worked with bacteria only that if you vaccinate it with bacteria orally and this vaccinia intravenously, do the two work together and the tumors go way faster or something. So this type of addition between bacteria and vaccine.
There's no doubt that this is a cure for many cancers, but the thing I always wanted to ask, I wanted to give the students opportunity, but with resistance for viral infections, such as like pVRNA, which is very much like differentially expressed in cancer, do you expect that maybe some tumors would be resistant to the therapy?
No, it's not resistant, but it is true that certain tumors need different concentrations. Therefore, dose escalation in preclinical trials, mouse trials is necessary. We always have to have one mouse trial where we add different doses to different cancers.
Just a little more for some certain types, yeah, probably to overcome that competitive inhibition that might hurt.
But you see it already a little bit in patent, it shows that some viruses are killing this tumor faster cells than some viruses. So there are some nuances, but not yes or no.
Yes, very cool.
No, it is like a metabolic symbiosis or something like that. Certain cells do have it, cancer cells supposed to have it.
It must be the glutamine.
Yeah.
It's the tRNA glutamine. It must be, and which is overexpressed in the cells. Yeah.
Sorry to pick up one question, which is when you go
Yeah.
to culture.
So my question: how many times have you heard this talk? And do you think that
Not
you
too
could
many
come and
times.
oh no. Maybe we need to have you come and speak and give this talk.
We do not know what we
I hear it
know
every
is
day.
that.
Oh, sure. Yeah, that's wonderful.
Because there are so many telephone calls that come from Germany, from his students.
And
It's
no matter life.
it's not
The
easy.
same is of course with CAR-T, and the same is
Germany
with
is 9
micro.
hours.
They are localized as
And the first call comes from when we
something is
then 11 p.m. when
the immune system,
11 p.m. when they are on their work day, there are Zoom calls and it goes for us from morning till night. These activities with overseas and
like the next published paper.
Well, that's a wonderful work.
Yeah, let's get some snacks and some refreshments.
Well, so thank you so much for coming and sharing. This is wonderful work you're doing. I'm Patrick Schott, chair of the chemistry department. And had to run the overflow session because there were so many people we couldn't fit them all in this room.
About 100
incredible
people you
talk.
didn't see. Yes,
Not a full 100 this time, but we packed this room and couldn't fit them all in.
Yeah, of course. Great to see you. Yeah.
It was supposed to be.
it's right behind. It's hiding. Testing, testing, very quiet, very quiet.
Test, test. Testing, test, test. Test. Oh, that's better. Testing.
Gotta make all three of these sound the same. This is your benchmark, okay? This is correct.
Testing, okay.
Those two are not working.
How do I do that back there?
No, you stay here.
Oh. Testing. Testing, 1, 2. What? Test, test, test. Oh. Test, test.
Alright, we have the tester online right here. Try it now.
Test, test.
Is it louder? Compare it to the green one.
Test, test. It kind of sounds like test. I think it's only coming out from one side. Test, test. It's lower. The blue one is lower. Yeah.
Turn two of them.
Test.
Turn two of them up. Did it get louder at all?
No. It got a little bit louder, but it's still low. It sounds like it's only coming out from... Okay. Test.
Okay. This is 7 and 8 back here. You see 6, 5, 1, 2. This is 3 and 4. 3 and 4. I just raised 7 and 8 was already like here at the top. Right? That's all the way right. 7 and 8. The 3 and 4, which are the only ones that in here. I raised to the same because they were turned like here. So I raised 3 and 4.
Are they able to go up anymore?
No, they stopped.
So they're all at the max.
So it must be cracked then. Must be turning off. Well, let me go. Because I know the issue too. I heard it the other day. How to paint it.
Whenever you're trading stress,
Can you yellow?
Huh? Oh. I don't think there's a battery inside. Also, this mic smells weird.
Check, 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 check. That's B. Check, check, check. It's louder.
It's good.
Check, 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 check. Check, 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 check. But I do agree with what she's saying. Check, check, check. A is only coming through on A and not B, and I think that's a Randy issue. He he made some changes recently, and at first the mics weren't even coming through, so he came and fixed them. He did some
Test, test.
So that's why they weren't at the same level.
Test.
I think that obviously there's still something like that going on.
Yeah.
Testing, test.
I remember when we...
I think it's people in the room will still be able to hear today, but...
Yeah, a long time ago. I think, I don't know if you were here yet, but it was with me, Carlos, and I think it was Micah Ramos when he was a little new. And we were trying to figure out a way to get these mics working in this room and these mics working in this room. Because even if you're in separate mode, the mics will go on both sides. So then we set it so that whenever you're in separate mode, the mics only...
That's how it should be.
That's how it should be, right? But
and then combined.
it had problems.
Yeah,
It wasn't
I don't
configured
think we should
correctly.
mess with any of that today.
Yeah.
But I agree, that's some of the stuff that needs to be looked at.
What? What? Oh, yeah. But it's not going to the other room, though.
This has been wonderful. We were going to see if we could place Oliver's bag or Dr. Salai's bag in here somewhere. Could you guys just make sure it's not being moved? I have my bag over here.
Check. 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 Yo, that's crazy.
Yeah, it's those little amp things. Yeah. Little amp. One was the one that he I don't know if he disabled them, and then he had to go. That's what the only reason I knew they were there.
I mean, it just has to do with the actual copper that's going, that's branching off. It's being swirled together, and then it goes to the separate spots. And
So
it's
you're
just,
actually turning, what,
You're
you're,
not
you're making
turning
more
anything.
contact or less,
You're
than,
adding,
than less
you're
contact?
adding copper to it, adding wires to where it goes, because it's the
So it has
actual.
a stronger signal?
Well, I don't think it's routed to both sides. I think both sides are separate. Remember how it was?
I was just talking about the volume on those little.
Yeah, I don't know. I don't know if turning up the volume will make it go on both sides. I think it's an actual.
No, not at all. But it was low.
Yeah, it was low. Yeah.
It was lower than the other.
Hello. Hi, everybody. Thank you so much for being here. This has been a great symposium so far. This is going to be the biological sciences oral presentation. So if you guys are part of the biological sciences, you're in the right place. If you're looking for a different oral presentation, please go and look in other classrooms across the way. That's where chemistry and mathematics and other departments are meeting. We're going to just go ahead and get right into this and get started. We're going to first start off with Emma Kamen and Tammy Stevenson. They're going to be talking about the dive reflex. I'm going to have them present for around 5 to 7 minutes, and then we'll have 1 to 2 minutes of questions in between each presentation. And so, yeah, we're going to go ahead and get started. Thank you guys for being here. You need a clicker, too. Let me get a clicker.
What if I were to tell you that our bodies have a survival instinct that can save your life? For instance, babies can hold their breath underwater for 10 to 30 seconds. You might be wondering, how is this possible? Well, it's because of the mammalian diving reflex. In our study of the mammalian diving reflex, we studied male and female non-athlete and athlete groups.
So what is the MDR? So MDR is a physiological response that happens with mammals when they get submerged under water and reduce oxygen. So what happens is that bradycardia occurs, which is slowing of the heart rate, and apnea, which is not breathing, and then vasoconstriction. So some similarities with aquatic and terrestrial diving mammals is that the heart rate decreases and vasoconstriction and apnea both increases. So the goal of our study is that we perform the MDR and observe the variation of heart rate among two groups: non-athletes, females and males, and athletes, female land and female and male swimmers.
For our methods, we started with an initial resting period. Our subjects rested for about 2 minutes, and we gathered their resting heart rate. Next, we did an acclimatization dunk. So our subjects dunked their face for 5 seconds, and then we also recorded this. Next, we went into the recovery period. So we waited until our subjects got back to their resting heart rate. And then the next part is the breath holding dunk. So our subjects dunked their face in the water until comfortable, and then this is where we recorded their highest heart rate and their lowest heart rate. In the post-dunk, we waited until their heart rate got back to their baseline.
So who did we study? We took 26 females, ages 18 through 22, 15 non-athlete females, and 11 female swimmers from CBU. We also studied 11 male swimmers from CBU and 9 non-athlete males. We recorded their drop value, resting heart rate, lowest heart rate, and highest heart rate. So our results concluded that for female non-swimming athletes, it revealed an average resting heart rate of 89 plus or minus 3 beats per minute, which decreased to 63 plus or minus 3 beats per minute during the breath holding phase. We also saw that for female swimming athletes, it revealed an average resting heart rate of 94 plus or minus 3 beats per minute and decreased all the way to 59 plus or minus 3 beats per minute. So our data demonstrated that the average heart rate across all subjects decreased to approximately 62 plus or minus 3 beats per minute during submersion. We can also conclude for male swim athletes versus female swim athletes that the pre-dunk heart rate measurements for non-swim, reveal an average resting heart rate of 67.09 plus or minus 2.45 beats per minute, which dropped by which dropped to 41.4 plus or minus 3.2% following immersion. Compared to female swimmers and male swimmers, heart rate dropped by a significantly greater percent, p equals 0.002. So the findings with our percent drop in heart rate showed that for female swim, which is pink, it showed a significantly greater drop percent rather than the non-athlete females, which showed around 26%, while female swimmers showed around 36%. Another of our findings is that for male non-athletes versus male swimmers, it showed that male swimmers in the blue had another great significant finding, which they had a bigger drop in their heart rate compared to non-athletes.
So like Tammy said, over here, oh, this one works too. So here are the non-athlete female groups. Here's the non-athlete male groups, the female swim, the male swimmers, and then we also have a land athlete group, and they were female volleyball players at CBU. And so this graph shows the comparison that male swimmers ultimately have the greater difference versus a non-athlete male, a swim female athlete, a non-athlete female, as well as a land athlete. And so this can further, we can further discuss our results later, but these results show that male swimmers have a greater mammalian diving reflex than the non-athlete and females. So after submersion, we asked our subjects a series of questions, and some had positive and negative effects. The positive being that they felt less anxious, relaxed, and then the negative effects, some were saying that the temperature was too cold to even participate, as well as they felt too anxious to participate. So, what can we conclude from this? Well, we found two distinct findings, which is amazing, and we found that athletes in general have a greater diving reflex, like in the big comparison, and then male swimmers have a greater diving reflex than female swimmers. In the future, we plan to research effects of the diving reflex compared to other athlete groups, as well as effects of age, anxiety levels, and then we plan to further investigate why these results from male and female are different.
And then this is where we got all our literature from. And then we would also just like to acknowledge Dr. Prinz for mentorship and also fundings from the Department of Biological Studies at CBU. Any questions?
Well done. All right, anybody have any questions?
I just want to ask, was age a factor? Did you see any difference between age groups or just gender?
We did not take a look at age. It was really just about athletes here at CBU. So it was between 18 to 22. So we didn't measure anybody below or above the age of 22. Thank you.
And I would like to add on that, we were born with this mammalian diving reflex. So we could see that the athletes are more trained to this diving reflex, and that's why it's more prominent versus looking at age.
So first off, great job, ladies, on the research. And then my question is, what would be an application of these findings?
Like an application, so we can use these studies to find.
Okay, great question. So as we research this, we see that athletes have a greater mammalian diving reflex than non-athletes. So what does that really mean? Well, it means that the athletes have a greater recovery than a non-athlete would. So that's what it shows in the mammalian diving reflex. So if you're more athletic in a daily routine, you'll have a better recovery that can ultimately help you in activity or swimming underwater.
Can I ask a follow-up question? Okay. Since you said that the males had a greater recovery, do you think that that has anything to do with lung capacity?
I believe so. We were talking about maybe getting a spirometer and checking lung capacity within swimming athletes to see if that does play a big difference. Because we were also looking at the data from the male swim athletes, and they were trying to see who can hold some were holding their breath for like 2 minutes in freezing cold water. So we think that it may have an effect in the mammalian diving reflex.
Well done. We don't have too much time for questions between each of these, so if you guys would give them a round of applause and say thank you for being here. Go run over to chemistry now. Yeah, we'll take that. All right, and then if the AV team, could you guys please switch back to slide number 2 for our next presentation? Excellent. Thank you so much. If Genevieve Estrada, Chloe Rogers, Leticia Sylvia Miranda, Sarah Rudd, Michael Alonzo, who are presenting up this next work, please come up. Thank you so much.
Either way, okay. Good morning / good noon, everyone. I'm Genevieve Estrada.
And I'm Chloe Rogers. Today we are so excited to share the results of our research on the molecular genetic manipulation of the receptor binding domain of the binary toxin for packaging into Bacillus thuringiensis, subspecies israelensis, or BTI, because that's a lot to say, Pilo.
Okay. So mosquito-borne disease continues to pose a significant global threat. Our research used an approach genetic engineering a novel bacterium that is highly toxic to mosquito larvae. So Bacillus, or BTI, as she mentioned, and Licinius Bacillus fericus, also known as LS, are widely used to control vector mosquito populations. Each species produces a distinct set of toxins that are highly specific that target mosquito larvae.
BTI and LS are both gram-positive spore-forming bacteria. So when BTI sporulates, it forms larvicidal protein toxins that are then trafficked into the pilo where they crystallize. Similarly, when LS sporulates, it forms this crystalline inclusion that is composed of the binary toxin bin-B, bin-A. So when the pilo or when the binary toxin crystals are ingested, they dissolve in the alkaline midgut of the larvae, and they're activated by proteases. Once activated, these toxins actually form pores that lead to the destruction, as you can see down there, of the larval midgut, and it just tears that thing up. And within a few hours, the larvae are dead. So this ultimately leads to a decrease in mosquito populations, which in turn leads to a decrease in human deaths from mosquito-borne diseases.
Yes, mosquitoes are famous for their very parasitic and transmitting viral diseases, such as dengue and malaria. These afflict many and hundreds of billions of people worldwide. And so now we want to see, what does BTI and LS affect the environment? So here we have Dr. Prince spraying biodegradable, eco-friendly BTI and LS. As you can see here, these BTI and LS are very specific to the mosquito toxin, so that makes it very only targeted mainly towards the mosquitoes, so it makes it more eco-friendly because of these receptors. Okay, so this also does do a reduces the negative consequences for chemical pesticides such as DDT, which is now used to kill mosquitoes, because once this is applied, it does stay into the environment for decades. So this would be a more eco-friendly approach.
So all of this leads us to our main question, right? How can we combine the LS binary toxin with the pilo to create an even more robust mosquito larvicide? So prior to this project, and you can see this right above, we were able to genetically engineer a strain of BTI. And this thing was really strong, really toxic, and it contained both the pilo and the binary toxin. However, you can see the two crystalline inclusions are actually separate in the cell. So what our work is focusing on now is creating a strain of BTI that's capable of trafficking the binary toxin into the pilo. This will yield a single highly toxic composite for delivery to mosquito larvae. So if there are two separate crystals, it's a lot less likely that the mosquito larvae would actually pick both up. However, with this strong composite such as this, you have higher likeliness and you have a much more rapid kill.
So how do we target the binary toxin into the pilo? This has not been proven yet, but we do think that there is a signal peptide sequence. As you can see here, the proteins are trafficked in a sequence alignment, and we think that the signal sequence is pointing to L sign on the N terminal of site 1AA toxin.
So to test this, we went ahead and we engineered two plasmids. One harbors genes coding for the wild-type binary toxin, and the other codes for a chimera. And this chimera is composed of the N-terminal site 1AA, and that includes the signal peptide sequence fused with bin B along with bin A. These genetic constructs were then introduced into a strain of BTI that doesn't typically form crystals.
As we can compare the control bacterium, the crystals were then, you can see on, I can't point, but right here you can see the control and versus the engineered strains. The engineered strains are producing crystals. We then performed an SDS page that produced the wild type BTI and as well as the genetically engineered binary toxin, which then led us to do bioassays. As you can see in the corner, we have the non-binary toxin, which is not toxic to Aedes, but that's because Aedes doesn't have the receptor for bin B. We also did as well as the, sorry, we also did the non-binary toxin to Culex, but they did die because of the receptor to bin B. Which was interesting was that the genetically engineered binary toxin did not die to Culex.
Yeah, so we were like, how do we explain this? So to explain this, we actually performed in silico modeling analysis, which helped us to gain insights to the 3D structure of the engineered bin B. So we know that natural bin B actually binds to the midgut via the N-terminal region before it recruits bin A. The 3D modeling, which you can see over here, the 3D modeling shows the peptide sequence from site A sterically blocks the N-terminal region of bin B before it can connect to bin A. So if you don't have receptor binding, you don't have toxicity. This is so disappointing. Or not, haha. So in previous studies, we were actually able to show that BTI site 1AA can synthesize activity of bin A without bin B or without amylo maltase from bin B. So this means that we can go ahead with the rest of our research to continue to get that bin B, bin A into the pilo.
So we introduced the recombinant plasmids into the BTI that has the pilo. We then compared the wild-type BTI that only synthesizes the pilo. BTI transformed to produce the natural binary toxin that shows two separate crystals. It also shows the engineered binary toxin showed some interesting results, such as a single larger crystal, which is more than likely pilo plus bin B plus bin A, and then two separate crystals in about 40% of the cells, and then also a larger crystal, which is likely pilo, which is attached to a smaller crystal in about 40% of the cells. The The results are very encouraging, and it seems like we're on the right track. So our bioassays, when we compared the recombinants to the control, we observed it increased toxicity. Interestingly, BTI produced pylo and the genetically modified binary toxin to about 6.7 times more toxic. And when compared to the wild-type BTI, that only produces the pylo. So this suggests that our results show that site 1AA provides a synergist effect.
All right, so for our future studies, we are seeing that the data that we're obtaining suggests that it is so possible to genetically engineer a stronger single composite pylo. So we went ahead towards the end and developed 3D models of a chimera composed entirely of site 1AA protein fused to bin B by a linker sequence with amino acid resides appropriately placed for flexibility between two proteins and for cleavage by mid-gut proteases, specifically trypsin and chymotrypsin. And we think that this construct will allow for the complete trafficking of the chimera with bin-B into BTI's pylo and for activation of these toxins when ingested by larvae.
Thank you all for listening. We would also like to thank the internal research grants for CBU for funding this, and as well as Dr. Badeshi for his mentorship and guidance. Thank you. And questions? No questions.
All right, do we have any questions? Oh, that got loud. All right, great.
So I know you guys were talking about how it's really non-toxic to humans. Why would that be beneficial, and like how would that factor into the future applications?
So we had mentioned that this dissolves in the alkaline midgut of the mosquitoes, right? Humans don't have an alkaline midgut. They have a super acidic gut, right? And so if you're spraying this out in nature, it's not going to affect humans because we're able to specifically target those mosquitoes. And that's really what we wanted with all of this. We wanted it to be healthy for the rest of the world. Yeah.
Time for, I think, just one more. Getting my steps in. Thank you, Stephen.
So kind of just to like piggyback off of that, are there any other, I guess, species or other animals, I guess, that also have an alkaline midgut that you guys would be concerned about, damaging?
No, because BTI and LS are so target specific to the mosquitoes, such as Aedes and Culex, that we don't really see that there would be a problem with any other animals or insects.
Excellent. Well done, you guys. So thankful that you guys gave that presentation. All right, I believe it is time for Kyle Yinger and Paris to come up and give their conversation about your work. Could we advance the slide one? Here, I can get the... There we go. Paris, there we go.
Before we start here, can I see, which one is it? That's back.
The green one will go forward, the red one's back.
What? Green one forward, the red one back?
Yes.
Got you. Here you go. Use that one?
Yes, please.
All right. Good morning and welcome, everyone. My name is Paris Luna, and today I'll be presenting our research on the in vitro generation of sgRNA, or small guide RNA, for gene editing in zebrafish embryos. To get started, gene editing is a powerful tool for studying biological processes. The CRISPR-Cas9 is one of the most influential technologies of this kind. However, there are some limitations when it comes to this research, as the generation of small guide RNA can be difficult and time-consuming. Traditional methods rely on cloning the sgRNA into a plasmid or DNA vector, which can be time-consuming. Our research presents a novel method instead to produce sgRNA without the process of cloning. The enzymes of the CRISPR-Cas9 complex work together to modify DNA and help us study gene function. The promoter drives this entire sequence in which the sgRNA guides the Cas9 to the DNA sequence of choice. Then the Cas9 nucleus actually cuts the DNA in which the promoter drives this, and we can modify that promoter itself to see how much complex we're creating. In order for us to target a specific gene, we need a customized sgRNA sequence. Traditional methods rely on cloning sgRNA to create a plasmid or a DNA vector, which can be time-consuming, complex, and limiting, as we can only do one at a time, and it takes about an hour for each. Cloning produces complex naturally, but we can instead purchase this and artificially assemble it. With this in mind, we sought out an alternative method in which we can create sgRNA without the need for cloning entirely. Traditionally, a plasmid is aligned under a specific promoter in which its expression is regulated. From there, the sequence is cloned onto the primer that's under, or the plasmid that's underneath it, and this is extremely time-consuming. Our method replaces cloning entirely. Instead, we use this and find our components by artificially linking primers with a T7 promoter, sgRNA sequences, and the overlap between the two respectfully. This creates a template that allows us to target multiple genes rather than just one. Does the clicker work on this? Okay, well, there we go. The laser?
Oh, the laser doesn't.
It's okay, no worries. Instead, our method aims to use polymerase chain reaction and in vitro transcription instead to replace cloning. PCR amplifies DNA templates in which we create our sgRNA by in vitro transcription directly. With this material, we're able to direct it into the zebrafish embryos early as 24 hours or just 15 minutes after fertilization. Microinjection of the zebrafish embryos allows us to model this in a biological model that's living. Zebrafish are the most ideal model for this research for a plethora of reasons. They have a very visual anatomy as they're almost transparent, as you can see right here. If it'll let me point, it won't let me point, but you can see up there that they're almost completely visually see-through, so that is very beneficial for observing under a microscope. And on top of that, they have very rapid development with major organs developing within just 24 hours. This factor is extremely beneficial as it allows us to see phenotypical changes within the first 24 hours. Microinjection occurs just within 15 minutes after fertilization, allowing us to interject in phenotypical development early on. On top of all of this, 70% of human genes have a zebrafish counterpart, making them a great biological model. In order to validate our method, we targeted a gene involved in eye development named OEP. Successful gene editing resulted in cyclopia, causing our embryos to grow just a singular eye. This is an X-linked recessive gene, so we were able to follow classic Mendelian genetics as we see our results here. On the left, we have our 15 control group in which they were uninjected, and we see that none of their alleles were altered. In the center, our 42 had one or none of their alleles altered, so they have no phenotypical change, although they were microinjected. However, those on the right show that we were able to change the zebrafish phenotypically, making them an OEP mutant and having them grow just a singular eye. This is extremely beneficial because it shows we can target multiple alleles and hit them, as well as it confirms the fact that we were able to generate sgRNA in this synthesis. It also confirms that OEP was necessary for the development of the eye. This is extremely important because our method does completely streamline the CRISPR workflow. As we're able to skip the cloning and vector stages, it broadens applications, allowing us to move at a much quicker rate. Although the plasmid is able to create it is able to create complex on its own. In vitro transcription cuts down the time and allows us to inject the pre-assembled complex automatically. This broadens our applications, allows us to apply this to other models, organisms, and disease pathways. This shows the effectiveness of our in vitro synthesized sgRNA for gene editing in zebrafish as well. Thank you so much to the Natural and Mathematical Sciences Research Symposium for having us, and to my team, Kyle Yinger and Dr. Zito, of course, and to California Baptist University as a whole. Any questions?
Well done, well presented. Do we have any questions for this work?
Yes. How much time does it cut back? So for the plasmids, it takes about an hour or more for just one round to happen. Whereas we do this, we're able to do multiple at a time, and at a much quicker rate. So it's not necessarily, there's not a definitive time, but it is more effective in the speed. It's more than half of the time. And then we are able to do so many more at the same time that it is more beneficial for us to do it this way. Thank you.
Any more questions? We got time for just one more.
Yeah.
Great.
How are we? What was that? For the knockouts, so we had the genome sequenced originally for these fish, and we were able to target that directly with the Cas9 generation that we had there. So it was just actually taking that genome sequence and then using our sgRNA sequencing technology that we have to do that.
Excellent work. Well done. If you have any more questions. All right, and now to follow up, Kyle in the same lab is going to come up and share. Thank you for being here.
I guess we could have just ended it off.
I know.
My bad. I led you astray.
It doesn't work.
The laser pointer doesn't work? Okay.
This one's got a laser, but that one's got the advance.
Try to do this all at the same time.
Yep.
Okay, that's back and that's forward. All right. Good afternoon, everyone. My name is Kyle Yinger, and I'm going to be presenting on our tagging and tracing technique for visualization of bacteria cells under fluorescent microscopy for furthering study in the host pathogen interaction. And we're going to be using zebrafish as our biological model for this. Bacterial infections and bacteria in general are just located all everywhere, and as Badeshi likes to say, bacteria is promiscuous and always changing. So building off of that, bacterial infections are a key component in the medical community and being able to address them and be able to trace them and the pathogen and host interaction that happens between them. Our technique of tagging and tracing is pertinent in furthering clinical understanding for bacteria interactions by able to illustrate and visualize in real time the interaction between the host cells and the bacterial cells at the same time. Let me see if I could get this. I don't know which one of these buttons. That would be cells inside, and I'm going to explain that in just a second for you guys. So you might be asking, how do we go about doing this? We go about doing this in two separate ways in our lab by doing fluorescent microscopy, by doing fluorescent labeling. The first way that we do fluorescent labeling is through fluorescent transformation, so direct transformation of E. coli cells. We use E. coli cells as our standard bacterial model. We do this by introducing foreign DNA via a plasmid and having it being incorporated into the genome of the E. coli. The plasmid contains a promoter and a sequence that then gets transcribed within the E. coli, producing our fluorescent proteins. The other way that we go about doing this is through a fluorescent dye. This is a fluorescent dye wash. The fluorescent dye wash is just like how it sounds. We soak the E. coli cells within the wash for a period of time, and the active component for it being uptook is going to be through diffusion. So from high concentration in the solution to the low concentration inside of the plasma, we watch as it goes through the cell membrane. The two main challenges that we were looking at when thinking about this project were how do we visualize our fluorescence how do we visualize it over the long term and under just how do we end up seeing this and tracing it.
The first way that we did this is our labeling systems had to be sensitive enough to be able to be picked up under UV visualization, and I'll show a picture of that with the ex vivo in just a second, along with being able to be seen under fluorescent microscopy. On top of that, the fluorescence also had to be stable enough to be able to allow us to trace the bacteria as it was going systemic throughout our biological model, and be able to have a half-life over longer than 24 hours, which I'll cover in just a second. As far as fluorescent transformation goes, fluorescent transformation, I told you guys it's kind of foreign DNA via a plasmid being introduced into the E. coli cell. As it gets introduced, it gets transcribed and then produced. Right here on the left-hand side of the screen is our GFP, which stands for green fluorescent protein within our E. coli. It's streaked out right here. This is what we were using. And then on the right side is an M. cherry, or a red fluorescent protein that we were using to also illustrate the fluorescence, to kind of have a duality between the two colors. We chose especially this method, because it helps with studying the gene expression within it. Also, because it is a fixed transformation inside and it's being produced within the cell, we wanted to be able to make sure that we could see the fluorescence coming from inside through the cell membrane. Then when it comes to fluorescent dye, we chose this method because it's very easy. It's not as time consuming. And right here, this is one of our samples. This was the FITC, or the FITC. This is a green fluorescent protein, or green fluorescent dye protein that we use. The other one we use is rhodamine B. It's a red fluorescent protein. I'll show you guys that in a little bit, in a minute, with some of the data that we saw. This one, these kind of dyes are mostly used for visualization of cell structures, viability, and microbial activity. So we really wanted to watch the study between the pathogens and the organism at the same time. That being said, you guys saw the pictures of how they looked under UV light. That's our ex vivo experiment. We were like, okay, cool, we can see the fluorescence. Now can we introduce these into our biological model to make sure that we can now see it underneath more layers, which means, like Paris was saying, the zebrafish embryo at 48 hours are transparent, or their melanocytes are not completely being produced just yet. So like right here in the diagram, they're pretty clear. We are going to introduce the bacteria via a direct inoculation through microinjection technique that I kind of teach here on campus, which is pretty fun. Virtually what we do is we take a micropipette tip and we inject directly into a few anatomical structures that are highly vascularized, one of them being the cardiac region, or the heart, or the pericardial sac. Moving down, we're going to go into the dorsal artery, which is right there in red, the caudal vein, and then at the very bottom, I don't think it's really highlighted, but the blood island. The biochem students, you guys know all about that. These are highly vascularized areas, which allows us to be able to mimic a bacterial infection as it's going systemic and causing sepsis within our biological model. That being said, this is a video of a proper injection that was done. If you can kind of see, I'm hoping you guys can kind of see it. Underneath the fluorescence, we see, oh my God, I clicked the wrong button. Not clicking the right button. So up here, this is the highly concentrated portion. This was a cardiac injection. We injected it into the heart. And if you can kind of see, if the video is still playing, yeah, I'm really shaky, I'm sorry. You can see the cells, the individual E. coli cells traveling through and also causing sedimentation throughout the vasculature and other organ systems, allowing us to give a kind of idea of kind of what's going on within the fish, and how giving us a disease dynamic about what's going on. That being said, this is the GFP, and I told you guys about the M. cherry earlier. This one's my favorite. It's a little bit more clear due to the difference in wavelength. The GFP has a little bit more artifacts, so finding out that this M. cherry was a lot more clear and being able to see the cells going through the vasculature a lot easier gave us the idea of, okay, what else can we do with this? What else are we seeing right now? This was a dorsal artery injection, so that's why you see the concentration kind of right there, where it's a little darker up here rather than down here by the heart. This is the heart and the eye down here, and this is all the vasculature going through. Next, so that was confirmation that we could see the transformation, the fluorescent transformation of our GFP and our M. cherry within the going systemic, and we can see it. Now let's try our other labeling technique, which was the dye. So right here, this is 2 pictures of our rhodamine B that we injected. These are cardiac injections. These are 2 separate samples. And we can see that the sedimentation, and we can see clear visualization of the E. coli cells right here fluorescing in a nice red view for you guys, all right? Now, we had the idea after that, what if we could do a co-injection? What if we were able to do an injection of our of our E. coli cells with the M. cherry that had been transformed inside, and then what if we wash them? Could we get a better visualization model and be able to directly confirm that we are seeing those E. coli cells in that exact area to give us proof about what's going on? Once we did that, we were able to look at it under UV light. This is a live image of one of our subjects. It's under bright field, and then we see it under GFP, and then we see the rhodamine right here, virtually confirming that we are seeing the sedimentation of these E. coli cells in the cardiac region of our fish, thus giving us a clearer image and enhancing our analysis of what's going on. In conclusion, our fluorescent labeling we've determined that fluorescent labeling enhances the study of our pathogen-host interaction by enabling precise tracking of infectious dynamics, allowing us to further understand like immune clearance, bacterial clearance, cell-to-cell mediation, and stuff like, and improving localization of pathogens within our host cells, thus providing insight into host immune responses. Based on our conclusion with that it works and we're able to see all this going on, we would like to do further implications that we see coming from that by establishing a fundamental basis based on testing it on other bacterial species to understand, like I said, a fundamental basis of the dynamic of infection of these diseases, to be able to outline exactly how that they're going to impact with their wild type, and then be able to look at instances of drug-resistant kinds of bacteria, or exactly how bacteria hide from macrophages. And macrophage, the word just lost me. Macrophage evading and immunochemical responses by dampening things like interleukins and complement proteins, and yeah, virtually hiding and what's going on within the bacteria itself versus this, and thus providing even more information for studying how the difference between a host and an infection or an infectious pathogen interact between each other. I'd like to thank the CBU in general and the Natural and Mathematics Sciences Department and the Research Symposium for having us here. Special thank you to Dr. Zito for all the aid and the encouragement in all of our endeavors, and Paris for always being there and being down to always do all the research. Any questions? Oh, I have to hold it, yeah.
Excellent work. I think those are the coolest pictures you'll see, is like the red bacteria flowing through. That's pretty incredible stuff to see. Very cool work. So anybody else have any questions about this research? I had one really cool. Oh, do you have a question? Okay, here.
How reliable is it to use glowing dyes in the E. coli injection?
Mm-hmm. As far as like a medical component and studying in humans, I don't have an idea of that. But for a biological vector, when we're using the fluorescent dye, we're seeing about a leakage or diffusion out of the cell after about 24 hours. So it does have a rate limiting step in that aspect. It's more of a confirmation that we use to make sure that we're watching within the transformation going on that would provide more information. Does that make sense? Yes. Anybody else have a question? Dr. Fuller?
If not, I do have one. So do you see any consistency in where these bacteria are going within the vascular tissues? Like, are they going and migrating? Obviously, they're going to go to the heart, but where else would they be depositing? And do they go there first? Are they consistently going that direction? You know,
right now, consistently with E. coli, we do see sedimentation very clearly in the arteries, especially surrounding the heart and in the heart. That's kind of where they're going.
For sure.
That's like the biggest portion. Yeah, we do need it. We want to do further studies than other bacteria for that exact reason, to study it further.
Is the gut fully developed at this point, or is it still developing
The gut is fully developed, but because we're avoiding the gut by doing just the vasculature. We're not seeing complete like diffusion from the gut into, or from the vasculature to the gut. If we're seeing what we're seeing as it leaks out, or as the fish passes the fluorescent dye into the water around it, we're watching as the macrophages break down and the dyes are leaking out of the cells already. Oh. Yeah.
Interesting. Very cool work. Thank you, Kyle. Round of applause. All right, and now we're going to have Sharon come up. If Sharon, there you are. Hi. She's going to share some of her research. Thank you. Use that microphone. Yeah.
This is the clicker and the laser pointers on the table.
Okay, thank you.
Presentation after this, I believe. Sorry, the one after this, the red one. This red one here. Yeah. Thank you, guys.
Hi. So let's talk about the reality of ovarian cancer. Unfortunately, 75% of patients experience a recurrence, and only about 30% of patients survive with a 5-year survival rate. Now, there are several mechanisms for the chemo resistance that these cancer cells face, and that's part of the reason that ovarian cancer is such an aggressive form. One of which being drug efflux pumps. Right here, they engineer proteins that actually form channels that pull the chemotherapy out of the cancer cell and prevent the DNA damage that it would have otherwise caused. That's just one of the mechanisms, but there are several there that cause chemo resistance. Now, this is how we wanted to tackle that issue. First, we created sensitive and resistant cell lines of ovarian cancer, and then we basically sequenced the genome or understood its expression levels of different oncogenes within that cancer cell line. We then completed RT-qPCR so we could quantify the gene expression of JAK-STAT proteins and IRDS genes, which we'll talk about in a minute. And RT-qPCR just takes RNA, converts it to DNA, and then uses that to measure expression levels. We then compared the expression levels between the resistant and sensitive cells, and we saw some pretty promising results. So let's get into it. Now, these are the various genes that I was referring to. Some of them are IRDS genes, some of them promote the JAK-STAT pathway. For example, LY6E is an IRDS gene or an interferon-related stimulated gene, and they just increase resistance. So the mechanism of LY6E is to induce stemness and increase EMT, which increases the metastatic potential of that cancer cell. Now, when we sequenced the genome or characterized the genes there, we saw that in the resistant cell line, there was an increased amount of TGF-beta and alpha, as you can see here. And then we also saw an increase in JAK-STAT proteins. So we wanted to study that pathway, and it really piqued our interest there because it was very unexpected. They also completed some enrichment plots and saw that interferon was highly regulated and released in these cells. So this is again an enrichment plot, and we compared the resistant and the sensitive cells, and we saw that the interferons were regulated and produced in the resistant cells when compared to the sensitive cells. Now here's the JAK-STAT pathway I was referring to. Interferons are usually released in response to a viral infection, so it's a very acute response that does not last very long. But when there is DNA damage accumulation as a result of the cancer cells being treated with chemotherapy, that causes the cell to be in a state of stress, which causes the interferons to be released more frequently. This chronic expression alters the pathway. So as you can see here, this part of the pathway is not present, and the STAT proteins are no longer phosphorylated. This change causes the expression of IRDS genes, those resistance genes, instead of the regular interferon stimulated genes. And we believe this is what causes the resistance in these ovarian cancer cells. So that is our hypothesis, that the DNA damage accumulation will cause an increase in IFN production, which will then cause an increase in stemness, metastatic potential, and so forth, therefore causing chemoresistance. Here's how we did this. We got ovarian cancer cells straight from patients from the hospital. We purified them, and then we extracted their RNA after creating a resistant and sensitive cell line. Once we extracted the RNA, we did RT-qPCR to quantify their transcription levels, and then we completed MTT assays. Those just verified that, yes, this is a resistant cell and this is a sensitive cell. So here are some of our results. PDX4 is a patient-derived cell line. We also worked with PDX6, so there's just two different kinds. This is the result of the MTT assay. We saw that the IC50, or the concentration of drug that was required to kill the cells, was a lot higher in the resistant cells and lower in the sensitive cells, proving that these strains are what we need to be when we move forward. So we then completed RT-qPCR on the various genes I mentioned before that are part of the JAK-STAT pathway and the IRDS gene signature. As you can see here, PLSCR1, IFI6, there was an increase in their expression levels in the resistant cells when compared to the sensitive cells, so that points in the right direction to confirm our hypothesis. When we look at interferon, we can see a difference here too that kind of tries to confirm our hypothesis. So here's PDX6 and other patient-derived cell line. Unfortunately, we didn't see very much of a significant difference here, so we moved forward with PDX4, and OVCAR8 was just done in sequence so that we could have a baseline moving forward, so it was used with data analysis. But with PDX4, since we did see a significant difference in gene expression, we decided to introduce it to ruxotinib, which is a JAK-STAT pathway inhibitor. And as I mentioned before, this altered JAK-STAT pathway is what drives the expression of the resistance signature genes. So when we blocked the expression of the JAK-STAT pathway, we can see that it sensitized the cancer cells to chemotherapy when compared to the vehicle, which is the control that was not treated with ruxo. So this does look like it's confirming our hypothesis. So in conclusion, we were able to show that the DNA damage accumulation caused chronic IFN1 signaling, which then leads to the expression of these IRDS genes, which will increase stemness, tumor plasticity, and overall therapy resistance. Now, in the future, we do want to explore some immunoevasive mechanisms that ovarian cancer uses, such as PD-L1, where it blocks T cells and evades the immune system. So we want to explore its relation to that. We also want to quantify IFN1 protein production by using Western blot, so we can show that the transcription levels really relay the same information as the actual proteins that are being transcribed. Then we want to disrupt IFN in the resistant cells to see if it sensitizes them, and then induce IFN in the sensitive cells and see if it causes them to be resistant, to really play with that one variable and verify our hypothesis further. But I do want to thank Dr. Julia Antonaire. She's been a great mentor at Loma Linda University, where I completed this research. I worked in her lab. And then Ms. Ashlyn Konart, who was an amazing PhD mentor to me. Then, of course, Dr. Dennis Badeshi, who's my faculty sponsor here that has been mentoring me for years. So thank you again. I'll take any questions if you have it.
Great work. Excellent project. Any questions about this? You can take one or two. All right, to seed it, I do have a question. So you've been working with the JAK-STAT pathway. Mm-hmm. I love that pathway. We look at IL-6 in activation of that pathway, but also we see it gives promotion of survival signals. Have you guys looked at survival signals, such as survivin or other proteins?
I don't think we looked at survival signals, but some of those IRDS genes do prevent apoptosis, so that I guess it's one form of survival.
That
right?
would be,
yeah.
I would say that. Excellent, excellent. Do you have any apoptosis assays you've run?
We didn't do apoptosis assays. Okay. This is very preliminary, but I'm sure Ashlyn is continuing it to verify the RTQ process.
Excellent. I'd love to see that. Anybody else have any questions?
Hi.
Hello.
Great talk. I'm just curious, when you are establishing the patient-derived cell line resistant to cisplatin, do you continue growing them in cisplatin, or is it just an initial concentration or dose and then you test it and leave it be?
Well, you kind of microdose it with cisplatin, so you'll gradually increase the concentration of cisplatin you apply to the cells, and once cells die, you kind of separate the live cells from the dead cells, and then you continue increasing the dose of cisplatin with the live cells until you culture like a resistant strain, if that makes sense.
Yeah, and you continue culturing them with cisplatin, because that's what we do with our pancreatic cancer cell lines, like they're consistently growing in gemcitabine. So do you do the same thing with ovarian cancer?
Yes, yeah.
Thank you.
All right, if that's all, thank you so much. Excellent, excellent work. All right, so if we could skip ahead in the presentations to now we're going to cover exosomes, another near and dear topic to my heart. So let's see, it's going to be Beatrice, Ivana, and Stephen, if you guys are presenting today. Come on up. We only have 2 microphones. Do you need them both? There, that's fine. Okay. Thank you, guys. Good. I'll make sure your presentation's up. So cool. Thank you.
Just real quick, just a volume test. Everybody can hear me? Yeah? Awesome. Okay, so start off, my name's Stephen Lyons.
My name is Ivana Kakish.
And Beatrice Santos.
And our research now is going to be exosomes triggered by Salmonella enterica. What are the macrophages communicating? I'm good.
I don't know what this is. Okay, first of all, we would like to thank our professors, which is Dr. Annabelle Flores and Dr. Ryan Fuller, for assisting with this project and to further do experiments with Salmonella enterica. Also, we would like to thank Kumaraswamy, which is a someone who helped us out last semester, and she is part of UC San Diego. And we also have a new person, which is Christoph Schroer, which is from Brown University, who is going to be assisting us with some further analyzing our results. Also, we would like to thank Mrs. Ali Floyd and Laura Floyd for setting up this presentation. And finally, we would like to thank CVU for helping us with our CVU research grant. Okay, Salmonella enterica. So it's important to know that Salmonella, it causes basically bacteria infection of approximately 1.3 million people yearly. And out of those, approximately 26,000 people get hospitalized due to complications from Salmonella. So Salmonella is antibiotic resistant. So it basically, we are analyzing the antibiotics and how they affect the Salmonella enterica. So some of the common symptoms with Salmonella is vomiting, diarrhea, abdominal pain, and fever. We're going to be seeing our strain, which is STO065. And yes. So media, why is media important? So media, we have two different types of medias. So CAMHB, which is cation adjusted molar Holber broth, is basically used in laboratory settings and at hospitals, and it's very popular. However, media should be important for human, to represent human conditions. Sorry. So the second one is the RPMI LB broth. So this media represents or helps grow mammalian cells. So it gives us more of a reference or a better way to grow mammalian cells, which is similar to our bodies. So it's important to choose our media while analyzing Salmonella when doing this experiment.
Let me use this one. Okay, so as Beatrice was saying, the media is important, and we want to highlight that within our assays. So in order to do this, we split our assay in half, having half of the media be the RPMI and LB broth, with it being 10% LB broth and 90% RPMI, and the other half being the MHB media. We then added the antibiotics, which was ceftriaxone, to our assay. And we started with the concentration of 64 micrograms per milliliter. And we serially diluted in half, going down the assay, going from 64 to 32, and so on going down. And having row G be our positive control and row H being our negative control. We then further divided the assay into quarters, having each quarter be a different strain. So we had one quarter being the strain ST065, and then the other strain being ST0539. And then we repeated that for both sides. This allowed us to test 4 variables at the same time and be very resourceful. So we tested 2 medias at the same time as 2 Salmonella strains. So based on these assays, we discovered that the Salmonella strain ST0635 was susceptible to about 32 micrograms per milliliter of ceftriaxone. And it was interesting because it was only susceptible in the RPMI media and not in the MHB media. So this further proves Beatrice's point that the media does matter, and RPMI media more accurately represents human body conditions, and it should be implemented more in hospitals. Okay. So furthermore, we wanted to apply this into human body conditions, and specifically in macrophages. So what is a macrophage? A macrophage is an immune cell that phagocytizes, so it will encounter a foreign body within a foreign element within the human body, and it will engulf it around itself, and it will dissolve it within itself until it is decomposed into easily disposed of pieces for the human body. And there's 2 different types of macrophage. There's the M1, which is the killer macrophage, which kills these foreign bodies, and there's the M2, which is the healer macrophage, which does more tissue repair throughout the body. And based on different environmental conditions, the macrophage can switch between these 2 different forms. And as the macrophage is in the body, it will transmit exosomes to communicate with each other. And these are just little vesicles with extracellular communication devices. And we're specifically focusing on the mRNA, which is inside the exosomes.
Okay, so before we hop into macrophages, I wanted to talk a little bit about monocytes. And it's going to be specifically Tohoku Hospital Pediatric-1 monocytes, or better known as THP1 monocyte cells. Now, this cell line was derived from a 1-year-old's peripheral blood, and this patient was diagnosed with acute monocytic leukemia a long time ago and has passed away, but their cell line still lives on, so we found it a little appropriate to give them thanks and appreciation, as our research wouldn't be, we wouldn't be able to do our research without their contribution to science. But going back to what Ivana was saying, we want to focus on stimulating our macrophages, or the communications between macrophages during Salmonella infection. To do this, we initially have to stimulate our monocytes into macrophages, and we do this by, I'm going to butcher this, fair warning, by using forbyl-12-mysterate-13 acetate. I'm going to call it PMA for obvious reasons. But yeah, so this works by activating a protein called protein kinase C, and protein kinase C is in a family of enzymes that work for cellular processes, mainly cellular differentiation. So once we administer PMA to our monocytes and stimulate them into macrophages, we would then go about infecting our macrophages with our Salmonella strain S20635. Now, this sounds very straightforward. We get PMA, we get monocytes, turn them into macrophages, add some bacteria, right? That's kind of what we thought initially. But once we infected our Salmonella, or our macrophages with our Salmonella, we completely forgot that bacteria replicates incredibly fast, right? So we ended up killing our macrophages very quickly and got some really awful pictures. But can we go to the next slide? But yeah, so once we get to that point where we do have our macrophages infected with our Salmonella, we really need to figure out a sweet spot in time where we're able to allow our macrophages to exhibit enough exosomes, which, how Ivana stated, is the communication between macrophage cells, and also not enough time to allow the bacteria to replicate to a point where it kills off all of our macrophage cells. But once we find that sweet spot, we plan to begin harvesting exosomes. And to do this, we already worked with Dr. Ryan Fuller and his research team. I told myself I wasn't going to mention it, but nice goatee, right? So he already gave us a lot of insight into how to extract these exosomes. he had a little background with exosomes, specifically harvesting some from E. coli, I believe. But one thing that he did tell us was that working with bacteria is super finicky when you're trying to centrifuge them. They work at varying times, and you have to change your centrifuge times and RPMs kind of accordingly. But once we work out this protocol, we plan to just take it through very low RPMs at a low amount of time, and then move forward to higher RPMs, higher amount of time until we get to that concentration of exosomes, where then we can then extract them and then send them out for analysis to our colleagues at Brown University. So the future applications of this, so the beginnings of our research was to fight or to contribute to the fight against antibiotic resistant strains of bacteria. But for the foreseeable future, we plan to push a lot of our manpower and women power to focusing on bioinformatics. So a lot of the analysis and data is going to come from Brown University, but we already have a team hopefully growing to break down all the information we're getting from this data. And from this, we hope to find a key insight into how these macrophages or how these cells communicate in response to bacterial infection. And that's going to be it, guys. Thank you.
All right, excellent work, guys. Any questions on the project here? We got enough time for a couple.
So for future studies, what kind of tests do you want to run for in terms of bioinformatics with Brown University?
As we were talking about, we want to analyze the mRNA pieces within the exosomes. We want to analyze those and see the difference between before the infection, after the infection, and potentially if ceftriaxone has an impact on these mRNA that is being transmitted.
And just to add to that, something that Dr. Fuller also mentioned to us, a lot of this is like unknown ground for us. So the questions and the research is going to kind of stem from what we find in that in the exosome vesicles.
Any other questions? Or else I'll have to keep talking. Hey, this is great work. I've got a follow-up. Now, recurring UTIs is a huge issue in the U.S. too, caused by similar problems with antibiotic resistance. Do you plan on expanding this study out to maybe bacteria that also affect that disease type? Or maybe you could speak to that a little bit?
Yeah, so again, it kind of stems from what we find from the bioinformatics side. But the ultimate plan originally was to branch this out into other strains of antibiotic-resistant bacteria. So it's really just going to stem from what we find, and then hopefully we'll be able to push it out to other bacteria strains.
Yeah, adding on to that, the ultimate goal is to see how do these bacteria become antibiotic resistant. And if we figure that out, then we can apply that to any antibiotic resistant bacteria. Well, potentially, that's what we hope, yeah.
Excellent. Do you hope to find some kind of sequencing output that has to do more with the bacteria or more with the macrophage response?
Yeah, the macrophage response is what we want to focus on.
Absolutely. Well done, you guys. Anything else? Oh. Apparently they were done, so just kidding. Of course, that was great. All right, let me get my list out here. We have a couple more presentations still to go. So could Evelyn please come up? Evelyn, you're up next. Thank you for being here. Everybody welcome Evelyn.
Oh my gosh, hi you guys, good evening. Thank you so much for joining me here today, whether it was against your will or not, still thanks nonetheless. Okay, so as you can see on the board, my presentation is going to be on Salmonella antibiotic resistant modulation in action through time kinetic assays. So quite the mouthful, but it gets all my bases nonetheless. Is it?
Next one.
The next clicker?
Yeah, that one works. Yeah.
Oh, okay. No. Okay. Awesome. So before we begin, I would like to give my acknowledgements to Dr. Annabelle Flores, Dr. Victor Nizet, Dr. Monica Kamaswamy, and then Miss Ali Floyd and other NMS faculty. I sincerely thank you from the bottom of my heart for allowing us to conduct this research, or just in general through your guidance and mentorship. Okay, a little background for you guys, a little fun facts, if you will. Okay, so Salmonella is a highly diversified pathogen with over 2,500 stereotypes identified. It's commonly found in poultry, eggs, reptiles, and it leads to food poisoning and typhoid fever. If I can bring your attention down to the bottom corner, that is an individual who's presenting with signs or symptoms of typhoid fever. The splotchiness, not very fun. That being said, my idea of fun may differ from yours. So, you do you, no judgment, only God can judge. Notably, Salmonella is a pathogen that can survive on surfaces for weeks, making it a very obvious, persistent public health concern. So in light of this, let's now shift our focus to the experimental procedure that investigates how ceftriaxone can address their challenges. A little, Q&A, if you will. That's not how that was supposed to go. Obviously, this is a sign that I shouldn't be up here. Okay, well. Go for it.
That one's just with a laser pointer.
Okay, this goes back.
That's back, yeah.
Awesome. Oh my gosh. It's missing the slide.
Is that on the border?
Yep. Ah, okay. Well, we're going to go out of order, a little keep you guys on your toes, see if you're paying attention. So allow me to walk you through my experimental procedure. First, we prepared a standardized inoculum of Salmonella 0539, which is the strain we worked with. Next, we introduced ceftriaxone to the sample. We then collected bacterial samples at time points 0, 2, and 4 hours. Each sample underwent serial dilutions, a series of serial dilutions, and then we plated them onto agar to count colony-forming units, also denoted by the acronym CFU, so keep that in mind. Finally, we compared the CFU counts at each time point against the baseline to understand the antibiotics' effect over time. So, before we get into our results, I forgot about this. Okay. Before we get into our results, I want to go back to the mechanism of ceftriaxone. So let's all take a trip down to imagination land. I know, you guys probably haven't been there in a while. We're all adults, unfortunately. So I would like you guys to all imagine that bacterium is a fortress with thick walls protecting it. Obviously, it keeps its shape. And these walls are built from tiny building blocks called peptoglycan. So normally, these special enzymes work like construction workers, linking these blocks together to make the walls strong and sturdy. So ceftriaxone is a beta-lactam antibiotic that acts like a saboteur, if you will, at the construction site. It stops these enzyme workers from connecting the building blocks. Without these proper links, the wall becomes weak, obviously, and it loses its shape, and then it dies. So that's called cell lysis. Very fun, very fun. And if you just tuned in, no worries, I got you. In summary, ceftriaxone doesn't allow the cell wall to be built. Awesome. So now let's dive deeper into our results. One more. There we go. So I would like you all to keep in mind this is very preliminary data, so don't judge me too hard. So our kinetic kill time assay, where we collected samples at 0, 2, and 4 hours, has shown that ceftriaxone begins the slow growth of Salmonella around the 4th hour mark. Although our charts are still being compiled, the early results align with our expected outcome. So we anticipated that the ceftriaxone would exhibit a delayed but a significant impact on bacterial growth. So data analysis is ongoing, but the trend observed at the 4 hour mark supports the hypothesis that antibiotic effects becomes pronounced after a short lag period. And obviously, I know this is not fair because I'm clipping, but so this is the 4th hour mark for the SM, which is denoted by SM, so it's just salmonella. And then the other one is Salmonella plus ceftriaxone, which is denoted by SM plus C. So if we compare the two SFUs, obviously the picture on the top it's in the 200s, obviously so many colonies. As opposed to the bottom one, it's like obviously decreased. Okay. We're going to do a little collaboration of sorts. So we're going to be referencing awesome Destiny Perez and her partner, Eric Hernandez. He's cool too. So the slide focuses on the interplay between our innate immune system and antibiotic treatment. The components of the blood, including red blood cells, white blood cells, like macrophages, and serum contain component proteins. All contribute to our immune response. Complement proteins are antimicrobial and play a crucial role in our innate immunity. So additionally, research is being led by the awesome Dr. Flores and actively working with macrophages to determine the precise role in the clearance of salmonella infection. This work aims to clarify the dynamic interplay between innate and adaptive immunity. Also, a little tidbit, plasma and serum are used interchangeably. They are synonyms. So if you're looking at that saying, that's not serum, Evelyn, that's plasma, just letting you know. All right. So our study suggests that there is a significant collaboration between innate immune systems and ceftriaxone in effectively killing salmonella cells, which is what we wanted. This can lead to further clinical findings such as dosing, although that does require more research because dosing is finicky because everybody reacts differently. And then as we saw or heard prior, things die really quickly if they're not in the right condition. Future applications. So what does this mean? So looking ahead, this has huge significant clinical implications. So the effectiveness of ceftriaxone against multi-drug resistant salmonella suggests a promising treatment strategy for infections. So our hope is to really hone in on these findings and possibly conduct future research on optimizing antibiotic therapy. Thank you so much for listening to my presentation.
Well done. Thank you so much. Any questions?
Hi. So
Hello.
you were saying that for the specific bacteria for salmonella, that there was that it was essentially antibiotic resistant. Have you done any research into if there's any specific antibiotics that are more effective than others, and what mechanisms are used to make them more effective?
To be quite honest, salmonella, sorry, let me remember your question properly. So you're asking if there's any other bacteria strains that is also resistant to ceftriaxone or salmonella that's resistant to antibiotics, I believe you said?
For the specific antibiotics that are more effective at destroying salmonella.
Got you. There's various antibiotics, but we were just specifically chose ceftriaxone because of its ability to, disrupt the cell building properties. So it just makes it the more better just because you can get inside like the actual cell itself and it can burst. In terms of other antibiotics, not to my knowledge, but I think I'd be really open to it just to see the different effects and then compare, do a baseline with the same strain at least to start.
Any other questions?
Can you explain a little bit more what you mean about stereotypes?
Of course. So stereotypes refers to the various distinction of species of bacteria. So as it just really talks about the specific set of antigens present on the cell surface. So like as mentioned previously, salmonella has 2,500 serotypes, and this is characterized by different antigenic profiles. Thank you.
I think we have time for just one more, if there is.
Awesome. Love you
All
guys.
right.
Thank
I do
you.
have a question though, really quick, actually, if I could ask it.
Okay, go
Seems like
for
we
it.
have a lot of salmonella growing on campus,
Of course,
right?
yes.
But in the labs, not in anywhere else, hopefully.
Yeah, for sure, yeah.
Now, I know we're excited about salmonella and everything. Where does it go in the body? When we get an infection, does it get into our gastrointestinal and then obviously cause all those symptoms? Does it migrate to our blood? How often might that happen?
Well, I think the most obvious one would be like it would migrate into your gastrointestinal, as you can see by like obviously like the food poisoning. So symptoms mean like vomiting and diarrhea, so a loss of fluids, but also you can see that it causes an autoimmune response. So you saw by the splotchiness for typhoid fever. So, but mainly I feel like in the gastrointestinal.
Yeah, and so it does kind of get out at one point and start becoming systemic.
Slowly, yeah. But mainly the focus would be like in that one point.
Yeah. And then you have that cytokine store.
For sure,
and all that stuff. Yeah. So cool. Excellent research. Thank you so much for presenting.
Thank you, guys.
All right, now we're doing great on time. Now, last but certainly not least, a lab that's very near and dear to my heart. You guys want to come up and start presenting? We figured we'd end on a big note, which is why we have everybody here. We only have 2 mics, though, so you guys are just going to have to make it work. Here's one. Here's 2. Good luck, you guys.
Hello, everybody. Thank you for being here. We're so excited to have everybody, and thank you for listening to us talk and share about all the fun things we get to do. We are the curcumin research team. We work under Dr. Fuller. Thank you so much. And we are researching how curcumin can affect pancreatic cancer cells. So, what is cancer, right? Cancer is a genetic disease that causes proliferation. And proliferation would mean the uncontrollable spread of cells in the body, right? But that's a basic definition. But how can we talk about cancer in a way where we can give it some characteristics that be more noticeable? And that's why we have this huge diagram right here called the hallmarks of cancer, or the characteristics. All cancers experience these hallmarks, but to different extents. And the two that we have circled are tumor-promoting inflammation and resisting cell death. The two we have circled are prevalent and more extent in pancreatic cancer. And on the diagram to the right, we mentioned 10 common cancers, and you can notice that pancreatic cancer is listed at the bottom with their relative survival rates, and because it has seen little to no change in the last 25 years. Like the reasons I've stated before, why is pancreatic cancer so bad? It is chemo-resistant. It resists the chemotherapy in the cells. And also the symptoms we've listed below, like stomach or back pain, or weight loss, or nausea, these are common symptoms which can lead to misdiagnosis and then a late diagnosis for patients, which can be even more detrimental in trying to fight the cancer later on. And we've listed many diagnostics, but the two I want to mention is about 8.5% of patients are alive 5 years after their diagnosis, which is a crazy statistic. And below that, I want to mention that it is currently the third leading cause of cancer death, and it is predicted to be the second by 2030.
The current treatments for pancreatic cancer include chemotherapy, and the two current chemotherapeutics are gemcitabine and FOLFIRINOX. Gemcitabine works by disrupting DNA replication, and it inhibits cell division, which causes the cancer cells to die. And then FOLFIRINOX is made up of those four main components, and it's used to treat pancreatic cancer cells that have metastasized. If we look over here at the longevity predictions, it is anywhere from about a year to 2 years, and that's with treatment. Slide. So then when we're looking for future directions, we are looking at curcumin as a treatment for pancreatic cancer. This is because curcumin is the active ingredient in turmeric, which is commonly used in traditional Asian medicine. And turmeric is often supplemented, as some believe it can be a treatment for arthritis, diabetes, and other diseases. The two main points that we want to talk about as curcumin as a treatment for pancreatic cancer is its anti-inflammatory properties and its anti-cancer properties. We've highlighted some of the anti-cancer properties below, as it induces apoptosis in these cancer cells and even in chemo-resistant cells. And that is why it is a promising treatment against pancreatic cancer cells.
Okay, so to do this, we used we used our cells, and we put them on cell plates. So when the cell plates would overgrow, we would passage them. So it's taking our existent cells and putting them onto new cell plates. So we had 3 different cell lines. We had PINK1, which is naturally resistant to chemotherapy. And then we had MIAPACA2, which is also known as MP2, which is naturally sensitive to chemotherapy. And then we had MIAPACA2 gemcitabine resistant, also known as MIAPACA2-GR, which was developed from our parental cell line of MIAPACA2, and this was with small doses of gemcitabine over about a year to create a high resistance to chemotherapy. Okay, so these different 3 cell lines represent the chemo resistance. So we, sorry. Okay, so we try to aim for about a 50% cell live count. So we used to do this, we used a 6-well plate, which we had 3 of the wells be our control with DMSO, and then we had the other 3 wells be our curcumin concentrations. So as you can see, we have our 50 micromolar of
curcumin, and this was treated with our pink 1, which all of our cells died. So then we tried this again with our 25 micromolar of curcumin on our MIA-PACA2 and MIA-PACA2 GR, and all of the cells died again. So we tried this again with our 10 micromolar for all 3 cell lines, and we saw that most of the cells stayed alive for our MIA-PACA2 GR and pink 1 cells, but the MIA-PACA2 cells did die. So from this, we can observe that curcumin limits cell proliferation and initiates cell death.
Okay, so to evaluate how curcumin affected and ensued cell death in these 3 pancreatic cell lines, we took live cell counts of each cell line 72 hours after treatment. So in this, in our first trial, we treated only panc 1 with a 50 micromolar of curcumin, and like Liberty said, we saw a really low live cell count at only 22%. Following this, we treated MP2 and MP2GR cells with 25 micromolar. We also saw way too much cell death at 19 and 13% cell viability. And so our last test, we only used 10 micromolar of curcumin, hopefully trying to get closer to that 50% live cell count. And so this was very interesting, seeing that MP2 cells were the most sensitive to this, and they had a 43% live cell count, followed by panc 1 at 70% and MP2GR at 93%, both of which these were more resistant strains. It's notable to look at the MP2GR graph on the far right. Between the 25 micromolar and 10 micromolar, we see a great difference in cell viability, and so it's likely in between these concentrations we found where the cells are resistant to curcumin. And then at the bottom, you can see a note that we performed an alternate t-test, which revealed the data for 10 micromolar treatment of GR and MP2 cells is statistically significant comparing between the reduction in both of those cell lines.
So from our research, we can draw several conclusions. First, we found that panc 1 cells are resistant to curcumin, MIA-PACA2 cells are the most sensitive to curcumin, and GR cells are unique in the sense that they were most resistant to curcumin at the 10 micromolar concentration. At 50 micromolars and 25 micromolars, all of our cells basically died. So it was really at the 10 micromolar concentration that we were able to see the cells expressing a difference, a noticeable difference in terms of their chemoresistance phenotype. And we plan to further test with concentrations between 10 to 25 micromolars to further explore that chemoresistance. But ultimately, these results revealed that treating pancreatic cancer cells with curcumin effectively inhibited cell proliferation and induced cell death.
Can we go one slide back? Back to the colonies. We also performed one forward. We also performed a long-term test, which Hannah was talking about the different cell resistance levels. And so we cultured cell colonies on these plates, and we're able to stain them with crystal violet. So you can see each purple dot is equal to one colony. And so we also used various concentrations for this test, and we mirrored the results as Hannah was talking about, our general results about the resistance of the cell lines.
Thank you. And then can you go 2 forward? Thank you so much. So in terms of future directions, we plan to test with more clinically relevant concentrations, such as those that would be used in treatments with patients. We want to further understand the mechanism of curcumin. We want to know if curcumin is effectively inhibiting receptors in the cell. We want to know if it is blocking the phosphorylation of STATs or the translocation of STATs as we've seen in previous literature. And we also want to explore the efficiency or efficacy of curcumin delivery inside the cell and see if pairing it with other treatments such as piperine or gingerol 6 or even exosomes would enhance the effects of curcumin in the cell. And that is our presentation. Thank you so much for listening. These are our references.
And we want to encourage you to
add some spice in the fight against cancer.
This is our research team, and we especially want to thank Dr. Ryan Fuller for his knowledge and encouragement and just guidance throughout this whole process and putting all these women together to do something so cool. Thank you.
Thank you.
Yeah, no, for questions,
Oh,
yeah,
yes.
yeah. Wow, what a great lab, right? All right, anyways, thank you guys so much for listening. Any questions on this particular project? And I can't speak on this one, so we need to come up with a few at least, yeah.
Great presentation, you guys. All right, a quick question. What concentrations of curcumin did you guys use, and how did you determine that concentration?
So for the different tests we use different concentrations. So for the shorter term 72 hour, we used 50 micromolar, 25, and 10. And so determining those, we had a stock solution, which was our 50 micromolar. And so we made that stock using curcumin and DMSO to like make it dissolve and make that stock. And so from the stock, for the rest of our experiments, we diluted it to whatever concentration we wanted to treat with.
Any other questions? Oh. They do have a caspase-3, yes. In fact, they have overexpression of survivin, which is causing a lot of that resistance. Sorry, I didn't tell you that. Now you know about caspase-3. Yeah. Yes. Yeah. Sorry, guys. Yes. Yeah.
So I'm just curious about kind of like the initial reasoning for, I guess, curcumin kind of starting off this whole research. Was there a difference in cancer rates? Because you guys mentioned that turmeric is used in Eastern or Asian medicine. Is there a difference in cancer rates between Asian medicine and, I guess, Western medicine or Western populations?
Can you hear me? Okay. So when we started coming up with this idea with Dr. Fuller, he kind of brought it up to us about curcumin, but then when we started researching it geographically, the rates for gastrointestinal cancers are lower in those Asian countries that implement more curcumin or turmeric in like their diets, right? So that's how we kind of came up with this hypothesis or this idea, and then how we want to continue to follow through with it, if that makes sense. So like geographically, yeah.
No. So the treatments that we were dealing with, especially starting at that high concentration of 50 micromolar, we would say are not clinically achievable because they're very high concentration. And so they would differ from like if you're supplementing with like foods containing turmeric in your diet.
Excellent. Any more questions? No, well done, guys. Thank you so much. This is so loud now. And that actually concludes our oral presentations. Thank you all so much for your support, for our students, for being here, for even your peers and your friends. I would like to invite all of you students and family members to go out and check out the grad fair that we have just outside. There will be various grad programs represented and things that you might be very interested in. They'll be able to talk to you about prereqs and how to get into their programs. So thank you all again, and we'll see you again soon.